ভিডিওটা অফ করে দিলে বেশি
অন করব না অফ করব?
অফ করে দে এখন।
হ্যাঁ সেটাই
আর মিউট করে দিস।
হ্যাঁ ঠিক আছে, অফই থাক।
জন্য নাকি?
Hello.
Hello. Yeah.
You can hear me, right?
Yes.
Okay. So Vikram, when we start, just let me know before we go live, then I'll switch on my camera.
Yeah, so we are live now. So at sharp 2:30 we can start the program.
Okay. So start के लिए I'll switch on my camera.
हाँ. Yeah.
Good afternoon, everyone. Namaskar. Thank you for joining us to celebrate NIBMG's 12th Foundation Day. Before we get into the program, we will join our hands in prayer for an invocation.
A very good afternoon to everyone. Our Director Sir, Professor Soumitra Das, Founder Director Professor Partha Pratim Majumdar, Professor Emeritus Sharmila Sengupta, respected faculty members, distinguished guest Professor Shiv Pillai, and the entire NIBMG family. I am A very good afternoon to everyone. Our Director Sir, Professor Soumitra Das, Founder Director Professor Partha Pratim Majumdar, Professor Emeritus Sharmila Sengupta, respected faculty members, distinguished guest Professor Shiv Pillai, and the entire NIBMG family. I am Aditya Agarwal
and I am Jonisman Sarma,
and we would like to take the honor of welcoming you all to the cultural program on the auspicious occasion of 12th Foundation Day of National Institute of Biomedical Genomics.
So it's been a tough time for all of us, and this pandemic has kept us away from each other for quite a long time. Hope you all are in good health. But the show must go on. That's why here we would like to have a flavor of vibrant cultural performances of the NIBMG family. So without any delay, we would like to proceed to the cultural evening of Foundation Day of National Institute of Biomedical Genomics. So, would like to invite Ms. Srija, Abarna, Madhurima, and Antara for the invocation of the ceremony. Over to you.
Thank you, girls, for a melodious rendition of our prayer. I invite Professor Soumitra Das, our Director, NIBMG, to give his opening remarks and welcome address. Sir.
Welcome to you all to this Foundation Day of this Institute, National Institute of Biomedical Genomics. As you all know, this institute, the journey started in 2009 when our beloved Parthoda, Professor Partha Pratim Majumdar, with the help of the then Secretary Dr. M.K. Bhan, they conceived and materialized this idea of establishing an unique institute which will address the biomedical genomics. The vision was to enhance knowledge, and at that time it was just a conception, but now you can realize that how the biomedical genomics can contribute to human health and disease. And the idea was to enhance knowledge and translate the knowledge using appropriate technologies for promotion of the well-being and the improvement of the genetics-based healthcare. So all this journey, I mean, we are just approaching teenagers. The institute have been doing with different genomes, the nuclear genome sequencing, mitochondrial genomic sequencing, metagenomic sequencing, and from last year we started doing viral genome sequencing to understand how the understanding of the genomics will help us to really or appropriately address the disease and help the mankind. So the institute over the years have generated a huge capacity for sequencing. We are now able to generate almost 192 human whole genomes per month, and we are targeting to reach 384 genome sequencing because of this powerful technology that we have established at the institute, and now it is run by the National Genomics Code, which is also supported by the Department of Biotechnology. So the idea is to improve the ability to diagnose illness, to classify the patient, to improve treatment. So something like precision medicine, which we are aiming for, reduce the public health burden and enhance the national health index. As I told you, that we have been doing it with the nuclear genome, mitochondrial genome, metagenome. But from last year, we were actually heavily involved in the national initiative when this whole country, whole world is stuck with this COVID-19. We try to understand how the genomics, how the genome of this particular virus is mutating fast and changing its character. Can we really understand it? So our institute is heavily involved in 3 different areas. Initially, we helped to set up diagnostics in different places, in different hospitals, some of the remote places, using our expertise and expert technologies, and helped the local hospitals to jack up the testing efficiency by providing skilled manpower and expertise. Then we actually initiated, with the help of 5 different DBT Institute, Department of Biotechnology sponsored institute, to aim was just to have 1,000 genome sequencing from different regions of India to figure it out whether the same virus or the same strains are actually behind the epidemic in the different regions, east, west, north, south. So we have had a huge network. I'm coming into that. And very recently, from March this year, we have started this Indian SARS-CoV-2 Genomics Consortium. Again, NIBMG is actually leading from the front along with NCDC, National Center for Disease Control, to handle this INSACOG, which I'm going to highlight it today. You might be hearing it everywhere in the newspaper, but probably for the first time, I'm actually sharing with our NIBMG family and the associates what is actually we have been doing it for last 1.5 years. Now, as I referred that we started this initiation of this process of sequencing with the 1,000 viral genome SARS-CoV-2, which was a pan-India, pan-DBT Autonomous Institute national initiative. We tried to sequence and try to find out what are the mutations that are actually affecting this and then spreading different parts, how fast they're spreading, when they are spreading, everything was actually mapped. And that gave us the strength and the confidence that actually we are now geared up into a much bigger problem. And we now have set up a consortium, which is initially was 10 national laboratories. Now we have expanded into 28, another 18 laboratories join hand with us. So this consortium set up sometimes officially in December 2020, the Indian SARS-CoV-2 Genomic Consortium. And as I show you on your right, you can see these are the institute which are actually instrumental in addressing these genomics questions, starting by NIBMG, then NCDC Delhi, then we have IGIB from Delhi, CCMB from Hyderabad, ILS from Bhubaneswar, INSTEM and CBS Bangalore cluster, CDFD from Hyderabad, NCCS, NIV from Pune, and also NIMHANS from Bangalore. What we are trying to address it here is to, first, initially we were trying to trace the international arriving passengers to somehow restrict the entry of different strains into the country. So we were trying to find out the international arriving passengers, if they are RT-PCR positive, trying to find out their sequences and try to connect it with their country scenario, duration stay, infection, their vaccination status, travel history, etc. We were also started routinely surveillance of the community samples. Different districts were actually now connected, and from every district we were getting certain definite number of samples. And we are also monitoring unusual scenarios and collecting more samples from those unusual surge places, and try to have this sentinel surveillance sites, which we call sentinel sites, supplying to this all these RGSLs, regional genome sequencing laboratories, the samples. And now we are actually trying to collect samples from hospitals also, which I'm going to show you later. And the idea is we are trying to have these international passengers, the community samples, and also the reinfected cases. You know, several people have been infected twice. Some people, now it's coming out even 3 times they were infected. We are trying to sequence all of them, whether there is mutations behind this repeat infection. And infection post-vaccination, even after full dose of vaccinations, single dose or full coverage, people are getting infected, whether there is any mutation behind that reinfection that we wanted to check it. And we are actually mapping. There are variants, you know, there are mutations in the virus can actually lead to something very clinically relevant. We call them variant of interest. And there are some of those mutations which actually causes of concern because it actually changes the virus property, the infectivity or immunogenicity, or sometimes it actually escapes our vaccine challenge. So we try to tag them as a variant of interest or variant of concerns. So we are actually identifying routinely from these sequences that which are all of variant of interest, which are of variant of concerns, and some of them could be variant of high concerns. And then we have a routine meeting every week. We meet with 2 different groups, and we try to have an internal discussion to find out how to strategize, and we inform government to appropriately change the policy if required. So the whole management plan is, as I told you, from districts, there are state surveillance officers. They coordinate with the district sentinel sites, collect samples, give it to all these 10 RGSLs. The sequences are done, and these VOC, VOIs are identified, and those informations are routinely shared with the National Center for Disease Control, and they try to share it in the public fortnightly. Some of these sequences are actually then uploaded into GISAID, which is an international flu database, and the samples which actually the mutations which are very relevant, and we found that it is actually triggering into a new sublineages. Try to isolate the virus in 4 different centers. One in Faridabad, one is ICMR NIV, one is in Hyderabad CCMB, and the other one is the Bhubaneswar ILS Bhubaneswar Institute of Life Science. Now, all these sequences are actually uploaded into 2 portals. Initially, it was in the IGIB portal, then NIBMG also have come out with the portal, and then these are mirrored. And we try to have a deep analysis of the sequence to somehow find out correlations with the mutations and some kind of outbreak or a particular something to worry about or a concern. As we map here, is like some of the mutations actually come and goes. It doesn't really accumulate. When it accumulates, you can see there are the color changes to deep color. And then we actually start looking at it, whether this mutation frequency is increasing, that has anything to do with the surge or a particular severity or a particular clinical scenario or not. So that's essentially, we try to trace it, We have been doing it, and IBMG is actually heavily involved in the analysis, and this whole team. I would like to thank in this opportunity the whole entire NIBMG team, starting with Arindam, Nidan, Sridhar, and all these newcomers, along with our seniors, Parthoda, and our national genomics centers. Genomics core is actually helping us to somehow cater this for the entire country. Now, along with this 10 national centers, we have now stretched to additional 18 centers and linked them into this hub centers. So there is a hub and spoke model. Now there are satellite centers along with the major primary centers, and they are trying to handhold the satellite centers to come up to the level of desired expertise so that they can have a quality sequence done, so that we can somehow spread and increase the number of sequences per month, and at the same time reach every corner of India to have a huge network covered. As you can see in this picture, this I borrowed from my friend Dr. Rajay Parida, and I just try to show you how these different centers are, different places are covering. And now we have added this 339 Sentinel sites are connected with the RGSLs with 28 sequencing centers spread all over India. And we now added 16 different hospital network. All the Apollo hospitals, AIIMS, and some of the local hospitals we are actually tagging in so that we can have sequences and the clinical information tagged together to have a real relevance to the study. And now we are actually planning to expand the scope of this INSACOG into SWED surveillance, and also trying to have more host genome sequencing so that we can try to have a greater or a larger picture behind the whole epidemic. As I told you, so the future of this INSACOG will probably try to expand it to the hospital network, to try to include the SWED surveillance, and we are in parallel trying to do some research to somehow correlate the impact of the mutations on the immunity in a longitudinal study to understand long-term post-COVID complications and the changes in the immunity setup. And we are also side by side trying to improve the infrastructure and the knowledge management so that all these informations are actually finally comes out to the public and helps in the human health management. So NIBMG, as I told you from the beginning, starting from the first report from Parthoda's group, where they showed that how D614G creates a havoc, and in the actually eventually you found that D614G in 2020 just overcast every other strength. But the first lead was actually initiated from NIBMG. Several studies are actually already published, and several studies are now in the submission procedures or under revisions. So as you see that this is a small way we are trying to, in a larger and a bigger initiative, to help the nation. But NIBMG, as I told you, we are just approaching 10. We are still growing. There is a lot to be done. We are doing it at NIBMG to help this COVID genomics project. And Dr. Kartiki Desai will also highlight what are the other academic achievements we had it and what are our new initiatives that are. I'll just try to give you a glimpse. These are the research areas. The institute have been doing it majorly in cancer biology. Now we want to stretch into cancer immunology and immunotherapy, and we are all looking forward to hear from Dr. Shiv Pillai, who is going to highlight in this area the recent trends and his research in this area. We are interested in the tumor heterogeneity and single cell sequencing. We're trying to have patient-derived 3D organoid cultures. You know, NIBMG's strength is the data science, and we're trying to analyze massive data sets using artificial intelligence and machine learning. We have initiated some research with the microbiome and genomic integrations. The crosstalk between the microbiome and the genome will help us to understand what is the larger picture for a disease progression. And finally, as I told you, that the viral genomics, which we have lately studied in from last year with the RNA virus, we were working with the dengue and hepatitis C. Now we stretched with the SARS-CoV-2, and we're trying to understand what are the implications of the viral infection in different hosts, in different individuals of different regions. And NIBMG is also side by side developing its the campus. The campus is more or less developed and clean. As you can see, I wish we could just meet physically and you could all enjoy the greenery in the campus. The buildings are all done, even the 3rd floor and the 4th floor. Many of you may not have visited. I think I would invite you to go, not in a group, but at least individually, maintaining distance. You can visit the 3rd floor and the 4th floor. Now it's ready for occupancy. And this is the right day to actually announce it. Now our 3rd and the 4th floor is ready for operations. And the faculty housing is also complete. It's already occupied by the 3 new entry to NIBMG family. And with this, I would like to thank, we are actually going through a dark patch, but we always believe at the end of the darkness, there is light. And NIBMG is actually showing the light. And I'm sure we'll come out of this dark soon. And with all your motivation, all I expect from my NIBMG family is little extra effort, little extra motivation and ambition. I'm sure that we have the ability, we have the strength, we have the confidence to reach to the light, and we can actually show the lights to the nation and the world. With this, I will hand it over to Dr. Anindam Moitra. Thank you.
Thank you, sir, for the beautiful introduction and your talk on the COVID sequencing. I would just like to add that reiterating what you said, that since the pandemic struck last year and it was declared as the international emergency of health emergency of international concern. We had been working round the clock on understanding the outbreak in terms of what we know best, which is the genomics of the SARS-CoV-2, and right from the beginning are the reports that we published from NIBMG, which predicted the D614G mutation, and subsequently we were the first report of the 9 sequences of SARS-CoV-2 from Eastern India that we did in collaboration with NICET and with IISc in understanding some of the implications of those mutations, was actually published as early as June last year, and has been one of the heavily cited publications on COVID from India of late. And after that, when we showed that there was a proof of concept, we were asked to lead the Pan-India 1000 SARS genome sequencing consortium, which you have already mentioned about, and we brought data from different states. So right in NIBMG, we sequenced the virus using a large network of clinical and other DBTI organizations from different states and showed how is the pandemic unfolding in various states of India and over a period of time. So the first about 2000 sequences of the SARS-CoV-2 genome were actually done and shared with the global community by this initiative, which was kind of the paving stone for the subsequent large-scale national sequencing surveillance program that INSACOG, which you mentioned, is actually right now conducting, and we are at the forefront of this. And so we are networking with a large number of centers. So primarily we are looking at West Bengal and each of the Northeast states in generating viral genome sequence data. So from each district of West Bengal and each of the far-flung states of Northeast, including Arunachal Pradesh, Nagaland, and Mizoram, from where earlier no sequences have been could be sampled, and also from Sikkim, we have, and as well as Tripura, Assam, and Meghalaya, we had been generating a lot of this sequence data and rapidly sharing with the global community for disseminating the results for so that can be looked into further depth and interpretations made on the outbreak. So, and as you mentioned that we are expanding the consortium. So I'm happy to say that till now, NIBMG has sequenced and shared 5,804 whole genome sequences of SARS-CoV-2 in GISAID. And of late, we have been more sequencing is ongoing in Northeast and West Bengal. So from West Bengal alone, we have sequenced about 3,933 samples. And from Northeast, all the Northeast states, till date, we have sequenced about 1,471 samples. And then again, whole other states as well, from Karnataka, Maharashtra, Haryana, Delhi, Uttarakhand, and Uttar Pradesh, and other states. We have also done some amount of sequencing. As you mentioned, that there are a few publications that have already come up, which deals not only with the viral genome, but the molecular epidemiology of the virus, as well as for the host genomic impacts. Some early insights have been published, and some more are in the preprints, and many more, I'm sure, will come soon. And I would just end by saying that now, in addition to INSACOG, we are happy to inform that our proposal, the STI framework program has been in the BRICS countries, have been funded by the BRICS STI framework program, where we have proposed to undertake SARS-CoV-2 network for genome sequencing surveillance in Brazil, Russia, India, China, and South Africa. In this international initiative, we are collaborating with University of KwaZulu-Natal in South Africa, with National Laboratory for Scientific Computing in Brazil, Skolkovo Institute of Science and Technology from Russia, and Beijing Institute of Genomics in China. And these are being processed in various stages of paperwork, and as such, this large multinational initiatives required, and very soon that study will also be on. In India, we are collaborating along with these countries with IISc Bangalore and CTFD Hyderabad. So I'm just saying all this as a part of a very large team, which includes Professor Das, Sridhar, Nidhan, a very large experimental laboratory team, where all the spanning across multiple initiatives, and also the team from the data analysis and the computational lab. So together we are working night and day to generate this sequence data for the national genome sequencing surveillance, and selflessly, rapidly sharing it in the public, so that not only us, but everybody can use the data to draw important inferences on the ongoing pandemic. I would take this opportunity to thank everyone who has participated in this, and which has been also supported by other academic members, and also the members from the administration and finance, who have tried to match us in so that we are not stuck in anywhere in generating this data in real time. I thank you very much, and thanks everybody for this splendid effort. Thank you.
Thank you, Professor Das. Thank you, Dr. Arindam, for giving us an insight into our newest baby, that is the INSACOG, and your brilliant achievements. What is tasked for me today is just to highlight some of the other academic programs that we run in the Institute. So please give me a moment to share my screen. And I hope you can see my slides.
Yes, you can see it full screen.
So, thank you. So I'm going to give glimpses of NIBMG's academic activities, and while doing academic, I'm going to throw in a few other activities that we do at NIBMG, which are a little bit fun too, so that you know about what's going on. So whenever we talk about a research institute, we generally think about performance indicators, which are these, right? Are the scientists present in the institute able to attract extramural funding? That means the scientific problems are well thought out, and somebody is going to give them the money to do it. So we have done brilliantly. Please ignore the 2020 and 21, as you know, because we suffered a pandemic. So obviously most of our resources have gone towards understanding COVID and its functions and its sequencing. But nevertheless, if you notice, NIBMG has performed so well in terms of its publications, which were on the rise in 2021, and also achieved a great citation index. A citation usually lets us know that if we have a good index in our field, many of the scientists are actually receiving our publications with great interest. Now, these are just basic performance indicators, but what really goes into achieving these year after year and improving upon them? So I thought I will just think of three things. It's not a comprehensive list, but definitely we need state-of-art facilities. And most importantly, we need bright young minds. They could be students, our postdocs, and also our proactive faculty, and a brilliant administration which supports all of these activities. And I think a campus that definitely rocks. So I'm going to share some things. Sir has talked about various achievements, and I would like to mention that if this was an Olympic, then we definitely deserve the gold, if not the highest medal, because our founder director, Professor Parthapratim Majumdar, way back took on the challenge to establish a state-of-the-art genomics facility. And they began with cancers of national importance, particularly with oral cancer, which was internationally funded. And because of that, so many of the publications have come out, and NIBMG was set on the map as one of the internationally renowned institutions for sequencing and also for understanding disease. So Sir dwelled a lot upon the health trajectories and artificial intelligence and so on. What I wanted to say was we need kind of a multi-platform approach for this. So I think a little bit of our present and a little bit of going into the future for a few years should be establishment of these facilities. So some of the cancers, I mentioned particularly oral cancer, and we are using cervical cancer under the leadership of Professor Sharmila Sengupta, who was also a director which is flagship project of NIBMG now, to understand how it comes about in healthy women and when the virus is cleared and why certain women actually sustain the virus for a long time, and ultimately it needs to presence of cervical cancer. And it's the flagship project of NIBMG, as I mentioned, and it will achieve a lot of data in terms of the understanding the microbiome, the proteome, metabolome, so on and so forth, and its integration with a lot of clinical data as well as 3D organoids and using liquid biopsy data. Now, in some of the areas, we actually find novel targets where we have identified them from the genomics data and we take it to the bedside. So these kind of experiments require significant validation and elucidation of the disease mechanisms. Now, even if we, particularly in cancer, even if we had to patent it as a biomarker or develop assays for it for any preclinical studies, we need to know the mechanism before we can file that patent. And this is what is attractive to an industrial collaborator later or a clinical collaborator in terms of helping you with samples and also taking your target into the drug discovery mode. The same case probably is true for complex and infectious diseases. We need significant validation and elucidation of disease mechanisms once we identify the host factors that are responsible for example, better or worse clinical outcome or more infection, or if we identify genomic lesion that is leading to a highly complex disease where multiple such losses or gains come together to give a complex disease phenotype. So for validation and elucidation of these disease mechanisms, we do require animal models, and for infectious disease, not only to culture the pathogen, but even to use the human samples, we need biosafety level 2 and biosafety level 3 facilities. And this is what NIBMG is now engaging, kind of establishing support mechanisms in which our genomic findings can go even further into the clinic, into the world, like we have successfully done for the COVID-19 virus. So we are just starting all these facilities, so please bear with us. Give us about 5 to 10 years. We are having a lot of teething problems, but we have established physically the proteomics facility, the biosafety level 2 and the zebrafish facilities, and in the pipeline are the BSL-3 and an independent microbiome lab. So soon we hope that as we go into the next few years, we are going to see more robust publication and fantastic citations when the papers start coming out, when these are combined with the original genomics data. So capacity building is nothing. All this is nothing. The facilities mean nothing if we do not have highly intelligent students and highly motivated individuals who can actually utilize the facilities. So we have a great capacity building and human resource development program at NIBMG, both which takes care of giving degrees to students and also training. So the PhD program, we already have 12 flag bearers, which we have proudly sent off into the world, which carry the NIBMG PhD name. Our students have reached beyond 40, but we would really like to take it more than 100 because we have such wonderful facilities now. So that is why we are engaged in expanding our numbers of PhD. What we realized in the PhD program is that if we can attract even younger students and give them an opportunity to learn something different and also learn how research is done and also give them some hands-on experience, it might attract them much more towards a PhD program. And with this in mind, we started the integrated MS PhD program a couple of years back. We have 17 students enrolled, and soon we will welcome a new batch since we just finished the interviews. So we do have a good postdoctoral program, which is funded either by extramural grants or national programs such as NPDF. We have a series of project trainees or fellows. We consider that they're all aspiring PhD students who are on their way gaining experience before they are going to join a PhD program, and oftentimes we encourage them to join us if they can achieve and get their own fellowship. The worst hit, I would say, are these two programs of NIBMG because of the pandemic, because our campus had to be restricted to members of NIBMG. Those are short-term training program and summer training programs for external scientists. Okay, but we are doing okay with that. So how do we go about doing it? We have a robust thing. We have to prepare the young minds for career in science and technology. Our motto is accelerating genomics for health. So how do we go about doing this? What we receive are applications and qualifications from students who come from at least 3 different backgrounds for their masters. Life sciences, computational, and statistical backgrounds. It is very important for us, for our PhD program to be successful, that these young students can talk to each other in a language that is understood by each one of them. That means we have to do a course, a special course design such that all 3 students are equipped with basic knowledge of what biology is, what computation is, and what statistics is. So we have a very unique a coursework program, which some of the highlights are presented here. I especially would like to mention the research methodology course, because that is the one which makes the students capable of designing a viable scientific problem and also teaches them ethics in research, which is very, very important. And the other is scientific writing, which is important because once you have good data, you should be able to communicate it to the world and help them understand what you have achieved. And as you can see, there are many other courses which bring them together. And last but not the least, we do have an exercise in which we can enhance our students' presentation skills in the form of a journal club, which is every Friday, and also help them give annual work seminars so that they can present their data for comments and input from the other research members of NIBMG. So that is all the official thing, but I think it's very, very important when you live on campus as students, because you know that Kalyani is really in a remote area. There is very little entertainment over here if you just go outside the walls of the campus, but that should not faze us. It should keep us up and about doing our research. So I think making the campus beautiful is really important, and we, in the form of faculty and administration, we have various committees which are looking into how to make this better. Okay, so we want the campus to be beautiful, young and vibrant, and we want to achieve more facilities for campus residents that may include faculty or students. So in the pipeline, we keep thinking about wellness and health of everyone. Maybe we can have a yoga center and many more extracurricular activities. One of them I have shown here. We probably should have a convenience store, a basic health center, or facilities for recreation, so there are social interactions that foster communication and exchange of ideas. We do try to grow our own veggies. We started our first NIBMJ badminton tournament last year, which got together a lot of people, and we hope the cricket tourney is revived. Excuse me.
The first performance for today is the song Tumi Ma Amake by the legendary Kishore Kumar. It was featured in the film Amarkanta. I would like to invite on stage Mr. Supriya Chatterjee for the performance. Over to you, sir. Thank you.
For your beautiful performance. The next performance is a recitation of the poem Nirarthak by Kavi Premendra Mitra. So I would like to request Samadrita for a performance. Over to you, Samadrita.
Thank you. অন্তরের ভাবকে রসের জাল দিয়ে প্রতীকের সাহায্যে প্রকাশ করাই তো কবিতা। অল্পের মধ্যে অতিশয়ের সংবাদ, প্রত্যক্ষের মধ্যে পরোক্ষের পরিচয়, নিকটের মধ্যে সুদূরের উপস্থিতি, নিরর্থকের মধ্যে অমূল্যের আবিষ্কার। আবৃত্তি করছি কবি প্রেমেন্দ্র মিত্রের কবিতা নিরর্থক। দরজা জানলা ভেজাও যত না, আকাশই তোমায় খুঁজবে। পাল্লা সারসি ফাটলে ফুটোয় কত কাঁথাকানি গুঁজবে। উঁকি দেবে দেবে দেবেই, যতই ভাবনা কিছু নেই, একদিন ঠিক শিরায় শুনিতে ছটপটে ছোঁয়া বুঝবে। যেখানেই কেন রওনা হও না, ঘরেই নিজে ফিরবে। তেপান্তরেও হারাতে চাইলে সেই দেয়ালেই ঘিরবে। ছাদে ঢাকা দেবে, দেবেই। যতই ভাবো না কিছু নেই, হিসেবে গোয়ালে আসরে বাসরে মামুলি ছক কে ছিঁড়বে। নিরুদ্দেশের পাল তুলে তবু নিজেরই সীমায় দুলবে। যেখানেই কেন উধাও হও না, প্রাণ তার বেড়া তুলবে। বেড়া দেবে, দেবে, দেবেই। যতই ভাবো না কিছু নেই, ছাড়া পেতে ছোট যেখানে খুঁটির বাঁধন কি করে খুলবে। যে ঘাটে কেন নোঙর ফেলো না, সাগর তবু ডাকবে। তরি ছেড়ে যদি তরু হও তবু ঝোড়ো হাওয়া ডালে লাগবে। নাড়া দেবে, দেবে, দেবেই। যতই ভাবো না কিছু নেই, ফসলের জল ঢালে যে সে মেঘই মোহমুদ্গর হাঁকবে। আকাশ তোমায় খুঁজবে, সাগর তোমায় ডাকবে। যতই ভাবো না কিছু নেই, একদিন ঠিক শিরায় শুনিতে ছটফটে ছোঁয়া বুঝবে।
Thank you, Shobhadita. That was a very beautiful performance. The next performance for today is the song Shravane Ghanai Tu Noyone by Nachiketa Chakraborty. It is based on the Megh Mallar Rag. I'd like to invite Shudipto on stage for the performance. Over to you, Shudipto.
Thank you. Okay. Nisare pa ma pa ni ra ni sa. Nisare pa ma ka re ni ra re sa. Srabono ghanai du noyone, srabono ghanai du noyone, আকাশের মতো আঁখি, আকাশের মতো আঁখি, মন বরিশরে শ্রাবণ ঘনায় দু নয়নে, শ্রাবণ ঘনায় দু নয়নে। Mapari dhari ni sa sa ni sa ni dhari ni sa 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 ni ni sa 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 বিষাদ বিধুর মনে তারি আনগোনা অধীর আকাশ বুঝি ছড়ালো সীমানা। বিধুর বিধীর আকাশ ছড়ালো সীমানা, অধীর আকাশ বুঝি ছড়ালো সীমানা। ছায়া ছায়া ভাসে ছবি, ছায়া ছায়া ভাসে ছবি, তার মন কোণে শ্রাবণ ঘনায় দু নয়নে, শ্রাবণ ঘনায় দু নয়নে। আকাশের মতো আঁখি, আকাশের মতো আঁখি, মন বরিশরে শ্রাবণ ঘনায়, শ্রাবণ ঘনায়, শ্রাবণ ঘনায়।
Thank you, Shalito. That was an excellent performance. The next performance is a dance on the Bengali version of the famous Odia song Rangavati. It will be performed by Pratyusha.
That was a brilliant performance, Pratyusha. So now I would like to request Mr. Devopriya for a recitation of the poem titled as Bharat Tirtha by Kaviguru Rabindranath Tagore. Over to you, Devopriya.
আমি তো ছিলাম ঘুমে তুমি মোর শির ছুঁয়ে গুঞ্জরিলে কি উদারত মহামন্ত্র মোর কানে কানে চলো রে অলস কবি ডেকেছে মধ্যাহ্ন রবি হেথা নয় অন্য কোথা অন্য কোনোখানে। রবীন্দ্রনাথ ঠাকুর রচিত কবিতা ভারত তীর্থ। হেমর চিত্ত পূর্ণ তীর্থে জাগরে ধীরে এই ভারতের মহানবের সাগর তীরে হেথায় দাঁড়ায় দুবাহু বাড়ায় নবীন নর দেবতারে উদার ছন্দে পরমানন্দে বন্দন করি তারে। ধ্যান গম্ভীর এই যে ভূধর
নদী জপমালা ধৃত প্রান্তর হেথায় নিত্য হের পবিত্র ধরিত্রীরে এই ভারতের মহামানবের সাগর তীরে রণধারাবাহী জয়গান গাহি উন্মাদ কলরবে ভেদি মরুপথ গিরি পটপথ যারা এসেছিল সবে তারা মোর মাঝে সবাই বিরাজে কেহ নহে নহে দূর আমার শরীরে রয়েছে ধ্বনিতে তার বিচিত্র সুর হে রুদ্র বীণা বাজো 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 ঘৃণা করি দূরে আছে যারা আজো বন্ধ আসিবে তারাও আসিবে দাঁড়াবে ঘিরে এই ভারতের মহামানবের সাগর তীরে এসো হে আর্য এসো অনার্য হিন্দু মুসলমান এসো এসো আজ তুমি ইংরাজ এসো এসো খ্রিস্টান এসো ব্রাহ্মণ সূচি করি মন ধর ভাত সবাকার এসো হে পতিত হোক অপবিত সব অপমান ভার মার অভিষেকে এসো এসো পড়া মঙ্গল ঘট পরে যে ভরা সবার পরশে পবিত্র করা তীর্থ রে আজি ভারতের মহামানবের সাগর তীরে।
Thank you, Devotlu. That was an extraordinary performance. The next performance is a song. It will be sung by Srija and Abarna, and the guitarist would be Animesh. So it's a fusion of two songs, Prithivita Naki Choto Hote Hote by the Bengali band Mohiner Khoragul and Bhigi Bhigi by James. Over to you guys.
Thank you.
Good luck.
পৃথিবীটা নাকি ছোট হতে হতে স্যাটেলাইট আর টেবিলের হাতে ড্রয়িং রুমে রাখা বোকা বাক্সতে বন্দি আহা হা হা আহা আহা হা হা আহা আহা হা ঘরে বসে সারা দুনিয়ার সাথে যোগাযোগ আজ হাতের মুঠোতে খুঁজে গেছে বেশ কাল সীমানার গণ্ডি আহা হা হা
Thank you, Srija, Aparna, and Animesh for your beautiful performance. Next, we are going to have a dance performance by Devarati and Somrita based on the song Tal Se Tal Mila. It has been composed by legendary A.R. Rahman and beautifully sung by all our favorite Alka Yagnik and Udit Narayan. So over to you, Devarati and Somrita.
Thank you, Devarati and Somrita. That was a very beautiful performance. The next performance is the song Sunri Pavan, sung by the legendary Lata Mangeshkar ji. It was picturized on Moshumi Chatterjee and Satyajit for the film Anurag in 1972. The lyrics were by Anand Bakshi and the music was by Sachin Dev Burman. I'd like to invite Antara on stage for the performance. Over to you, Antara.
सुनरी पवन पवन पुरवैया मैं हूँ अकेले अलबेली तू सहेली मेरी बन जा साथिया सुनरी पवन पुरवैया मैं हूँ अकेले अलबेली तू सहेली मेरी बन जा साथिया तू बन जा साथिया चल तू मेरा चल थाम के अनजाने रास्ते से गाँव के चल तू मेरा चल थाम के अनजाने रास्ते से गाँव के साथी है ये मेरे नाम के गोरे किस काम के डोले मेरा मन ऐसे जैसे नैया मैं हूँ अकेली अलबेली तू सहेली मेरी बन जा साथिया तू बन जा साथिया कोई तो हो ऐसा पूछे बात जो गिरू तो पकड़ लेवे हाथ जो हंसे रोए सदा मेरे साथ जो सोए जागे संग दिन रात जो ऐसे हूँ मिलन जैसे धूप छैया मैं हूँ अकेली अलबेली तू सहेली मेरी बन जा साथिया तू बन जा साथिया
fabulous performence. The next performance is another recitation. I would like to invite Mr. Ayan Majumdar for recitation of the poem Africa by Kavi Guru Rabindranath Tagore. Over to you, sir.
নমস্কার। বিশ্বকবি রবীন্দ্রনাথ ঠাকুরের কবিতা আফ্রিকা। উৎক্রান্ত সে আদিম যুগে স্রষ্টা যখন নিজের প্রতি অসন্তোষে নতুন সৃষ্টিকে বারবার করছিলেন বিধ্বস্ত, তার সেই অধৈর্যে ঘন ঘন মাথা নাড়ার দিনে রুদ্র সমুদ্রের বাহু প্রাচী ধরিত্রীর বুকের থেকে ছিনিয়ে নিয়ে গেল তোমাকে। আফ্রিকা, বাঁধলে তোমাকে বনস্পতির নিবিড় পাহারায়, কৃপণ আলোর অন্তঃপুরে। সেখানে নিভৃত অবকাশে তুমি সংগ্রহ করছিলে দুর্গমের রহস্য, চিনছিলে জলস্থল আকাশের দুর্বোধ সংকেত। প্রকৃতির দৃষ্টি অতীত জাদু মন্ত্র জাগাচ্ছিল তোমার চেতনা কিন্তু মনে। বিদ্রুপ করছিলে বিষণকে বিরূপের ছদ্মবেশে, সংজ্ঞাকে যাচ্ছিলে হার মানাতে আপনাকে উগ্র করে বিভীষিকার প্রচন্ড মহিমায় তাণ্ডবের দুর্দুহীন নাই। আয় ছায়াবৃতা কালো ঘোমটার নিচে অপরিচিত ছিল তোমার মানব রূপ। উপেক্ষার আবিল দৃষ্টিতে এলো ওরা লোহার হাত করে নিয়ে, নখ যাদের তীক্ষ্ণ তোমার নেকড়ে বেঁচে এলো মানুষ ধরার দল। গর্বে যারা অন্ধ তোমার সূর্য হারা অরণ্যে যে সভ্যের বড় বড় লোভ নগ্ন করলো আপন নির্লজ্জ অমানুষতা। তোমার ভাষাহীন ক্রন্দনে বাষ্পাকুল অরণ্য পথে পঙ্কিল হলো ধূলি তোমার রক্তের অশ্রুতে মিশে। দুষ্টু পায়ে কাঁটা মারা জুতোর তলায় বিভৎস তালার পিণ্ড চিরচিহ্ন দিয়ে গেল তোমার অপমানিত ইতিহাসে। সমুদ্র পারে সেই মুহূর্তে তাদের পাড়ায় পাড়ায় মন্দিরে মন্দিরে বাজছিল পূজার ঘণ্টা। সকালে সন্ধ্যায় দয়াময় দেবতার নামে শিশুরা খেলছিল মায়ের কোলে। কবির সঙ্গীতে বেজে উঠছিল সুন্দরীর আরাধনা। আজ যখন পশ্চিম দিগন্তে প্রদোষকাল, যখন ঝঞ্ঝা বাতাসের উত্তশ্বাস, যখন গুপ্ত গহ্বর থেকে পশুরা বেরিয়ে এলো, অশুভ ধ্বনিতে ঘোষণা করলো দিনের অন্তিম কাল। যুগান্তের কবি আসন্ন সন্ধ্যার শেষ রশ্মিপাতে দাঁড়াও ওই মানহারা মানবীর দ্বারে, বলো ক্ষমা করো হিংস্রতাপের মধ্যে সেই হোক তোমার সভ্যতার শেষ পূর্ণবাণী। নমস্কার।
Thank you, Ananda. That was a very good performance. The final performance for today is the song Rote Hue Aate Hain Sab by the legendary Kishore Kumar, and it was featured in the film Mukaddar Ka Sikandar. It will be performed by Mr. Tamogna Chatterjee. Over to you.
रोते हुए आते हैं सब, रोते हुए आते हैं सब, हंसा हुआ जो जाएगा वो मुकद्दर का सिकंदर, वो मुकद्दर का सिकंदर जाने से जीता जहां प्यार से जीते दिलों को झुका दे आसमान जो सितारों पर कहानी प्यार की लिख जाएगा वो मुकादर का सिकंदर वो मुकादर का सिकंदर जाने मन कहलाएगा रोते हुए आते हैं सब हंसता हुआ जो जाएगा जिंदगी तो बेवफा है एक दिन ठुकराएगी जिंदगी तो बेवफा है एक दिन ठुकराएगी मौत महबूबा है अपने साथ लेकर जाएगी मर के जीने की अदा दो दुनिया को सिखलाएगा ओ मुकादर का सिकंदर ओ मुकादर का सिकंदर जाने मन कहलाएगा रोते हुए आते हैं सब हंसता हुआ जो जाएगा हमने माना ये जमाना दर्द की जागीर है हमने माना ये जमाना दर्द की जागीर है हर कदम पे आंसुओं की एक नई जंजीर है सारे गम पर जो खुशी के गीत गाता जाएगा वो मुकादर का सिकंदर वो मुकादर का सिकंदर जाने मन कहलाएगा रोते हुए आते हैं सब हंसता हुआ जो जाएगा वो मुकादर का सिकंदर वो मुकादर का सिकंदर जाने मन कहलाएगा जाने मन कहलाएगा जाने मन कहलाएगा
Thank you, Tom Mukunda, for the beautiful performance. And that was the end of today's cultural program. And we hope that you liked all the performances.
We both, on behalf of the organizing team of Foundation Day of National Institute of Biomedical Genomics, would like to thank you all, and also the performers who made this event a spectacular and vibrant one. We'd like to thank you all.
We would also like to thank all the viewers for their patience and time. Have a nice evening.
Thank you. Since this is the month of August and India will soon be celebrating 75th Independence Day, so here we would like to conclude the program with a patriotic song by Dr. Souvik Mukherjee. He will be singing the song Dhano Dhanyo Pushpo Bhora by D.L. Rai. I would like to request Dr. Souvik Mukherjee to come on the stage. Over to you, sir.
Thank you. ধন্য ধন্য পুষ্প ভরা আমাদের এই বসুন্ধরা তাহার মাঝে আছে দেশ এক সকল দেশের সেরা ও সে স্বপ্ন দিয়ে তৈরি সেজে স্মৃতি দিয়ে ঘেরা এমন দেশ আমার জন্মভূমি। চন্দ্র সূর্য গ্রহ তারা কোথায় উজ্জ্বল এমন ধারা, কোথায় এমন খেলে তড়িৎ এমন কালো মেঘে, তারা পাখির ডাকে ঘুমিয়ে ওঠে, পাখির ডাকে জেগে। এমন দেশ কি কোথাও খুঁজে পাবে না গো তুমি, সকল দেশের রানী সেজে আমার জন্মভূমি, সেজে আমার জন্মভূমি, সেজে আমার জন্মভূমি। এত স্নিগ্ধ নদী কাহার, কোথায় এমন ধূম্র পাহাড়, কোথায় এমন হরিত ক্ষেত্র আকাশ তলে মেশে, এমন ধানের ওপর ঢেউ খেলে যায় বাতাস কাহার দেশে। এমন দেশ কি কোথাও খুঁজে পাবে না গো তুমি, সকল দেশের রানী সেজে আমার জন্মভূমি, সেজে আমার জন্মভূমি, সেজে আমার জন্মভূমি। পুষ্পে পুষ্পে ভরা সাখী, কুঞ্জে কুঞ্জে গাহে পাখি, গুঞ্জরিয়া আসে বলি পুঞ্জে পুঞ্জে ধেয়ে, তারা ফুলের ওপর ঘুমিয়ে পড়ে ফুলের মধু খেয়ে। এমন দেশ কি কোথাও খুঁজে পাবে না গো তুমি, সকল দেশের রানী সেজে আমার জন্মভূমি, সেজে আমার জন্মভূমি, সেজে আমার জন্মভূমি। ভাইয়ের মায়ের এত স্নেহ কোথায় গেলে পাবে কেহ, ও মা তোমার চরণ দুটি বক্ষে আমার ধরি, আমার এই দেশেতেই জন্ম যেন এই দেশেতেই মরি। এমন দেশ কি কোথাও খুঁজে পাবে না গো তুমি, সকল দেশের রানী সেজে আমার জন্মভূমি, সেজে আমার জন্মভূমি, সেজে আমার জন্মভূমি। ধন্যবাদ।
Thank you.
Thank you to all the students and the cultural team. That was a really wonderful show. Let's move on to doing what we do best, science. So we have a series of talks by 3 young faculties and 2 senior PhD students in store for the first scientific session. Beginning with, I would like to welcome Dr. Arvind Korwar, who is the assistant professor and has joined recently. And he will be having a lecture on the topic mass spectrometry based proteomics and metabolomics.
Hello, am I audible? Hello, good afternoon, everyone. I thank the organizing committee and the director of NIBMG for giving me this opportunity to present my research experience with you all. So today I will be talking about my experiences with the mass spectrometry based proteomics and metabolomics. So today I will give a brief explanation about my research background and the central research theme which I would like to continue at NIBMG, and the research topics which I would be considering as long and short-term goals or interests do research at NIBM. So coming back to the research background, throughout my career I did mass spectrometry based proteomics and metabolomics. Basically, I started my career as a project assistant with Dr. Mahesh Kulkarni at CSIR NCL Pune. Then I did my PhD at from Pune University, followed by I was CSIR RA also with Mahesh Kulkarni. For my PhD, I worked on breast cancer clinical tissue samples. Title was Molecular Profiling of Breast Cancer for Targeted Therapy, a Proteomic Approach, wherein we deduced the network signature for luminal A subtype of breast cancer, as well as we profiled the metabolite signature for luminal A subtype of breast cancer using NMR technology. And for the first time, we showed the glycated proteome in clinical breast cancer tissue samples. Then after that, most of my research at Dr. Kulkarni's lab was associated with identification, characterization, and quantification of special molecules called advanced glycation end products in murine and human clinical plasma, especially associated with the diabetes or diabetic complications. Then I moved to Singapore and worked with Professor Z, wherein I worked on plasma proteome, wide characterization of post-translation modifications, including carbamylation, acetylation, etc. And I was heavily trained on two important quantification workflows called PRM and MRM workflows. Then I thought I should work more on wet labs, so I moved to US and started working with Dr. Sandeep Prabhu at Penn State University, wherein I was introduced to biology in a great deal by all the lab mates at Penn State at Dr. Prabhu's lab, wherein I investigated the role of selenium, which reverses the Warburg effect in polarized bone marrow derived macrophages during resolution of information, and also developed a comprehensive MS-based method to quantify selenoproteins in a cell lysate. Then I moved to Center for Translational Biomedical Research to work with Dr. Kevin Zhang, who is from PNNL, Washington. There I was trained thoroughly as an analytical biochemist, wherein I had a great deal of access to high-end mass spec in the market right now, wherein I worked on implication of metabolome, lipidome, and glycated proteome in the development of cardiovascular complications in type 1 diabetic subjects. Let me briefly introduce you to advanced glycation end products. Predominantly, we see hyperglycemia in diabetic condition, wherein the concentration of glucose is very high in blood plasma. That excess glucose interacts with amine group of proteins and forms initially SHIP base, then amide product, which is reversal. Then they tend to undergo condensation reactions to form stable compounds called advanced glycation end products. Predominantly, carboxymethyl lysine and carboxyethyl lysine accounts for 80% of this advanced glycation end products. Nevertheless, we do find mold, gold, and DOLD sort of interaction, which cross-link two different proteins. So that induces the aggregation of these proteins and form protein aggregate. And these are associated or involved in various diseases from diabetes to Alzheimer's disease. So here I am jumping to one new story, wherein I would like to explain the application of proteomics at clinical setting, wherein we studied the albumin role and the levels associated with increased plasma protein glycation and HbA1c in diabetic setting. If you look carefully, the albumin concentration in diabetic high and diabetic low plasma samples, wherein you see the higher level of HbA1c and the lower concentration of albumin also, which suggests that whenever there is decrease in the albumin levels, that tends to undergo glycation, thereby protecting the other low abundant plasma protein. So the same is manifested and studied by two different aspects. One is two-dimensional gel electrophoresis as well as in solution digestion, that is shotgun proteomics. So wherever we see the diabetic low albumin plasma, there is higher degree of glycation of plasma proteins, and the same was orthogonally supported by Western blot analysis for few of the proteins. So this suggests that albumin protects other low abundant proteins from glycation. I'm jumping to another story wherein we developed the diagnostic fragment ion library for glycated peptides in human serum albumin. It's very important to study the peptides of human serum albumin because it is slowly been considered as another marker for monitoring glycemic index in other than HbA1c. So wherein we briefly synthesized the glycated albumin by incubating the human serum albumin with glycolic acid or methyl glycol or free glucose to get a carboxymethyl lysine modified glycated peptide or carboxyethyl lysine modified or fructosylysine. So by doing that, we subjected all these different glycated human serum albumin to in solution tryptic digestion, which were acquired on high-end Orbitrap mass spectrometer. Then we also established the diagnostic fragment for amalgam modified lysine, carboxymethyl lysine, and carboxyethyl lysine modified glycated peptides of human serum albumina. And we also validated these diagnostic fragments by using targeted PRM workflow, which again to establish, again we use this built-in library and validated with diabetic clinical samples, wherein we propose these 13 different types of peptides which could serve as a signature to monitor the glycemic index in diabetic subjects other than HbA1c. Then now I am jumping on to another story called selenoproteins and their implication in resolution of inflammation. Selenocysteine is a 21st amino acid which is encoded by UGA stop codon and is designated by letter U. And serine, cysteine, and selenocysteine, they fall in the same group when it comes to when in periodic table. And these selenoproteins, there are totally 25 in number in mammalian and 24 in murine. And predominantly the most famously known are glutathione peroxidase and thyroidoxine reductases. The biosynthesis of selenoproteins is a little bit complicated, wherein sec-tRNA is coded by a special gene called TRSP, and then we have a Sekis binding protein, which is very huge complex, and the biosynthesis of selenoproteins is still contradictory, and there are many things yet need to be known. I am showing this slide for the fact that I had the privilege to work with TRSP knockout and SBP2 knockout models in the Penn State lab with Dr. Sandeep Prabhu. The idea is to see that the role of selenoproteins in different pathophysiology. So that one of that fact that I want to study with respect to inflammation. So the current paradigm of resolution of inflammation is whenever you have insult or injury, you have edema, followed by a gush of neutrophils, then followed by macrophages to clear the area. That leads to resolution. And during this initial onset, lipoxin, resorption, and prostaglandins play a very important role in setting up the resolution. Whenever there is any disturbances during this onset, that leads to uncontrolled resolution or failed resolution, which leads to chronic inflammation. And initially, this resolution of inflammation process was thought to be a passive one, but Dr. Professor Charles Serhan was the first one to coin it as an active process, and he especially works on a specialized pro-resolvent pro-mediator. With that background, I was thinking what we can do with respect to inflammation when it comes to the macrophages. So metabolic phenotype of M1 and M2 like macrophages is well defined and well known. M1 metabolic phenotype is predominantly glycolytic, which is Warburg effect, and it is exactly similar to the cell cancer cells. In case of M2 like macrophages, it is predominantly ACPOS, which is also known as alternately activated. So, considering the M1 phenotype of the accumulation of succinate is hallmark of M1 phenotype. So that leads to and that is a pro-inflammatory phenotype. So whatever we do, if we bring down the succinate levels down, that will trigger the phenotype and predispose us towards pro-resolution phenotype. So far, CARKL, which is a carbohydrate-like kinase, which catalyzes the orphan reaction in non-oxidative phase of pentose phosphate pathway, where it converts pseudouptose to pseudo 7 phosphate, is well known, and it plays a very bigger role in predisposing M1 phenotype towards M2 phenotype. Similarly, pyruvate kinase is also well studied and well known. Similarly, succinate dehydrogenase is also well studied and well documented in literature. With this background, we hypothesize that selenoproteins regulate metabolic reprogramming in macrophages that predisposes these M1 phenotype towards the pro-resolving phenotype, that is M2 macrophages. So with this, we designed the experiment such that bone marrow derived macrophages were cultured for 7 days in presence or absence of sodium selenide, and they were treated with stimulated with LPS, and cells were harvested at 0, 4, or 8 or 28 hours after the stimulation, and the sample was extracted and subject to 2 different platform technologies, that is metabolomics and proteomics. We did a targeted metabolomics wherein we looked for 43 different types of metabolites, and we were basically interested in the metabolites which belong to glycolysis or pentose phosphate pathway or TCA cycle. However, we saw the dramatic changes in the metabolite levels, usually at 4 hour and 8 hour, and each tube represents biological triplicate, and we have selenium treated and selenium untreated cells. We saw a dramatic change in the conversion of succinate, wherein in presence of selenium, the succinate levels came down dramatically when we compare to the cells which were not treated with the selenium. Then our proteomic data also suggested dramatic changes in the whole proteome. But apart from that, we specifically looked at the proteins which belong to TCA cycle, and we also see the dramatic changes or upregulation of this succinate dehydrogenase enzyme specifically at 4 and 8 hours after stimulation with LPS. And the same phenomena we didn't see with the knockout mice model, suggesting that metabolomics and proteomics orthogonally tested with Western blot, all the data corroborates. Further, we also tested this phenomena in vivo using Zamasin A induced peritonitis model. We treated this mice with DMM, dimethylmethylate is a competitive inhibitor of succinate dehydrogenase, and after the treatment we collected peritoneal exudates at 0, 12, 24 and 48 hours, and we did the flow of neutrophil flow, and whenever we see the double positive of F480 and CD206 which is macrophages of M2 phenotype, they were more in DM untreated cells, suggesting that the DMM treatment negatively impact the selenium dependent resolution of inflammation at least by 20 hours. So in summary, in wherever you see in red color, they are induced upregulated, and the green ones are downregulated. So whenever we have the presence of selenium in the form of sodium selenate, there is an channelized TCA cycle, which is because of the activation of succinate dehydrogenase, and this changes the whole phenotype of the macrophages from M1 phenotype and predisposes towards pro-resolving phenotype, that is M2 macrophages. So at NIBMG, this would be the central theme of my research interest. Proteomics has different wings when it comes to the basic studies, including structural proteomics to proteome mining. But specifically, I'm interested and I'm trained in two different platforms, that is protein expression profiling and post-translation modifications, which I would be pursuing throughout my career at NIBMG. And these are the long-term and short-term goals as long as I'm at NIBMG. The first one is regulation of metabolomic or proteomic remodeling by selenoproteins in the context of chronic inflammatory diseases or disorders, including cancers and diabetes. And the second one is the regulation of pathways of inflammation and the resolution in immune cells, including neutrophils, macrophages, by micronutrient called selenium. Though I'm not a trained immunologist, but I'm trying to learn immunology, but I'm more interested in immunometabolism to study the effect of micronutrient on resolution of inflammation. And lastly, deciphering the genetic basis for the development of diabetic complications. If we have two different subjects, one is only one develops the diabetic complication and other doesn't. So I'm more interested in studying the genetic basis for the development of diabetic complications. Thank you. I would take this opportunity to thank our director for providing funds to establish the metabolomic facility at NIBMG as well. Thank you.
Thank you, Dr. Arvind. Next up, we have Dr. Anup Majumdar, who is an NIBMG fellow here, and he'll be talking on deciphering the novel mechanisms of host pathogen interactions to combat infection.
Is that okay? Am I audible? Okay. Good afternoon, everyone. So it's a great opportunity for me. I'm Anup Majumdar. I have recently joined in NIBMG, and it's a great opportunity for me to present my scientific career so far with all of you, and that entitles like deciphering the role of novel mechanism of host-pathogen interaction to combat infection. So I started my scientific career in PhD with microRNA in Dr. Subhendra Bhattacharya's lab of IISc Kolkata, where I worked on how this microRNA regulates the immune response, the host innate immune response. So you know that the microRNA is a small regulatory RNA molecule that can be derived from the primary microRNA and can bound to the Argonaute protein and can form a RIS complex. And then this RIS complex bound microRNA can bind to its target mRNA and can replace the translation. And this bound microRNA bound mRNA can be stored in a T body or processing body, where it can be either degraded or after stimulation it can be again de-replaced to go into the normal translation. So that way this microRNA replication and this thing is reversible. And I was interested to see how this microRNA regulate different cytokines during the bacterial lipopolysaccharide infection model. So LPS, bacterial lipopolysaccharide, LPS, is a TLR4 like. So when it binds to the TLR4, it can activate different downstream signaling molecules. And it was already known that the different cytokines like IL-6 or TNF-alpha, IL-10, these are all highly regulated by different microRNA, and also the TLR4 pathway itself. So when a cell, a host cell or macrophage cell, interact with any bacterial lipopolysaccharide, so if there is a huge expression of different cytokines, but still microRNAs are there. They are repressing their expression. So think about it. We are accelerating simultaneously. We are applying the brake. So I think this is not feasible for the cell. So there must be something so that the cell can induce this induce the transcription of the cytokines freely. So that was my question, like how this cytokine mRNA become immune to microRNA in activated macrophage. So first, I check the microRNA activity with this LPS stimulation by dual luciferase system, where I transfected with this granular 3xB site. It's a luciferase reporter that has 3 LET7A microRNA binding site. So when I transfect this, the LET7A host cellular LET7A microRNA can bind here and can replace its translation. In this way, we can get the full repression. So that will represent the microRNA activity. So when I measured this microRNA activity with LPS stimulation for different time points, surprisingly I found that there is a decrease in microRNA repression. That means there is a small time window when microRNA becomes inactive, so that the cytokines and all these things can be translated freely. So that makes sense. Like in the initial time window, when we need a lot of cytokine expression, so microRNA becomes inactive for a while, so that cytokine can be expressed very freely. So that was our very interesting observation. Then we look how it's happening. And there is another interesting observation that these are the P-bodies where microRNA can reside, and with LPS stimulation, the AGO2 positive P-body number decreases. So that means the AGO2 bound microRNAs are coming out from the P-body. And then there was a report from other group, the Gunter Meister group, that the Argonaute protein can be phosphorylated at this 5 to 9 position. And this form a binding pocket, so where the microRNA can bind. So if there is a phosphorylation at this pocket, then the microRNA comes out and this interferes with the microRNA binding. So I thought, yeah, this is the reason for our case also. So I checked the microRNA and this AGO2 phosphorylation level with LPS stimulation and found that there is a reversible increase in the AGO2 phosphorylation at initial time point, and it reverts back to the later time point. And also with this, I found that with the initial period, there is a decrease in microRNA association with the Argonaute protein. So from this, we conclude that, okay, with LPS stimulation, so there is a tyrosine kinase becomes phosphorylated, activated, and it can phosphorylate the Argonaute protein. And this Argonaute phosphorylation interfere with the microRNA binding, so that microRNA becomes inactive for a while, and so that cytokines and all those things can be translated freely. So that means this adds another layer of regulation in the cytokine lineage. So this was the project one for my PhD, and there is another interesting project that was I was doing simultaneously with the Leishmania infection. So as you know, some of you know that Leishmania is a obligate intracellular parasite that can reside inside the macrophage and that can evade our immune response and can harbor inside the macrophage. But how this Leishmania can evade the immune response is not known completely. So that was another thing that I was always thinking, that how Leishmania can evade the immune response. So when I was doing this LPS stuff, then another thing I found that whereas LPS increasing the Argonaute phosphorylation, but with the Leishmania infection, there is a decrease in Argonaute phosphorylation. That means because Leishmania always want to evade the pro-inflammatory immune response. So think about it. So if Leishmania wants to do so, it will always try to repress the pro-inflammatory cytokine. That means if it can reduce the, if it can dephosphorylate the AGO2 level, so that the microRNA repression will be always there. And in that case, the pro-inflammatory cytokine will be replaced. So that was the reason behind it. And I found, okay, yeah. So since Leishmania is replacing this or decreasing this phospho-AGO2 level, that means the repression is always there, and that helps the pro-inflammatory cytokines at a minimal level. And then I thought, okay, what are the phosphatase? That means there is some phosphatase. Leishmania is inducing some phosphatase inside the cell after infection that can reduce this phosphoagouti level. And it was already reported there are different phosphatases like PP2A or SHP1 and PP1. So those are activated by Leishmania infection. And then by this siRNA study and some other pharmacological study, I found out that the PP2A, this is the phosphatase. Leishmania becomes activates the PP2A phosphatase, and that can reduce the phosphoagouti levels inside the cell. And later there's another protein called HUR. It's a very important protein that regulates the. It can bind to the 3' UTR of many different mRNAs, and it's a RNA stabilizing protein. And also, HUR has a different role in the microRNA regulation pathway. It can act as a microRNA sponge and many other things. And where I also found that this Leishmania infection, the HUR level goes down at the later time point. So that means Leishmania is also regulating HUR protein. So later, the other colleagues from our lab, you have found that they described a tripartite regulatory axis involving this PP2A, AGO2, and HUR protein in the regulation of this pro-inflammatory anti-inflammatory cytokine response with the Leishmania infection. So that means there is a complex regulatory pathway is working with this microRNA pathway, and this way Leishmania can establish a secure infection inside the macrophage cell. So this was the infection part. Then I became interested about this transcription machinery, how it's happening. So I moved to UCSD in Professor Gauri Shankar Ghosh lab to work on the non-canonical NF-kappa pathway. So there is a most of us know the canonical NF-kappa pathway, but there is another non-canonical NF-kappa pathway where the RELB and P52 is the active dimer. So in the unstimulated cells, there is a this RELB and P100 protein becomes inactive. But with stimulation, this NIC-IKK kinase complex becomes active, and it can process the P100 into P52. And this RELB and P50 protein can becomes active from heterodimer. It can enter into the nucleus and can activate different transcription events. So, but how this NIC-IKK kinase, and there is a RELB, that's the degradation enzyme, it can degrade the P100 protein. So these two opposite proteins, the NIC-IKK1 and the RELB, has opposite function. But how this, with the stimulation, how this process happens was not completely known. So that's why we looked for it and found that with the stimulation, so there is a competition between the NIC-IKK complex and the RELB to bind for the P100 protein. And the relative concentration of the NIC protein or the RELB protein finally determines that which one will be the final outcome. From like different biochemical assays, we have found that like there is a transient complex formation between the P100 protein, RELB, and NIC-IKK complex. And based on the concentration of either NIC-IKK or the activation of rather NIC-IKK and RELB, so it will determine whether it will go for the P100 processing into P52 or whether it will form an inactive kappa B formation. So that was the story about the NF-kappa B. But there are other different transcription factors that is also known at that time. And I was thinking like how in the immune response, how these different transcription factors can be activated or how they are dynamically regulated. Means we know that some genes are regulated by different transcription factor like IRF3, NF-kappa B, and AP1. But how combinatorily they are controlled was not known that much. So then I moved to Professor Alex Hoffman Lab of UCLA to understand the quantitative understanding of this transcription factor dynamics in stimulated macrophage cells. So as I mentioned, like there are different terms, means it can activate the macrophage cells, lipid, TNF-alpha, poly-A are different. And with this different stimulation pattern, their different transcription factor will be activated. But their activation is dynamic, means not always all the transcription factor will be active, or the amplitude of this activation is also different. Like sometimes the NF-kappa will be more activated compared to IRF3, or AP1 will be more activated compared to NF-kappa, like that. So, but when we see, we just tell that it is activated. But how much activation is there, or how they are combinatorially controlled is not known properly. So that was our question, like how whether we can stick or whether we can say somehow at how they are combinatorially controlled. For that, we thought, okay, we will generate a lot of experimental data, and from that experimental data, we'll try to build a computational model. From this computational model, system biological model, whether we can predict that whether how these transcription factors are dynamically controlling different vaccine genes. So for that, I checked like with different camps, we take the RF3 activation or NF-κB activation or ethnion activation, like that. By phospho-RF3 is a mode of activation of RF3. When the RF3 gets activated, it gets phosphorylated and can go to the nucleus and can bind to different genes. So by this wisdom of the different stimulation, I quantified that how much activation is there. However, their activation amplitude was different. Like with LPS, activation was more compared to lipid A or compared to polyase. So I have to normalize also the activation pattern, and in this way, I got like a lot of quantified data for which we can put into our model. And also like this is the MSA for NF-κB for different stimulation like TNF for LPS for different time points, where I can see once they are activated, they can bind to the DNA, and from this we can get the activation signal. And also like for different stimulation, we can get this type of signals. And when this transcription factor binds, they transactivate the gene. The transcription happens. But to ensure that what are the nascent transcripts that we're getting, I did a chromatin, other type of RNA-seq experiment that is called chromatin associated RNA-seq or cRNA-seq. So from here we can measure the nascent RNA so that we can directly correlate this nascent RNA transcription with different activation of transcription factors. Then combining this transcription factor activation data and the cRNA-seq data, we put into our model, and we generated an iterative mathematical modeling approach. We took an iterative mathematical modeling approach based on this and the logic gate to build the model from where we can predict that how much activation is there with the different camps and how much transcription will be happen for different cytokine genes. So this was the this transcription factor activation and the dynamic control. Then I was also interested about another recently discovered non-coding RNA that is called enhancer RNA. So I joined this Shannon Lovett lab in UCSD to check that how this enhancer RNA can modulate different oncogenes in the cancer, colon cancer system. So it is already known that there is a cis-acting DNA element that is called enhancer. So this enhancer may be far away from the gene promoter, but in some way they are regulating the gene transcription. But previously it was known that like this enhancer form a chromatin loop, and in this way some transcription factor can bind to the enhancer, and in this way it can regulate the gene transcription from the promoter. But, so there it is known from all the like 30-40 years ago. But in 2010, there after this invention of this genome sequencing, people have found that scientists found that there are many short transcripts. Those are found from the enhancer region. Previously, it was known that maybe those are transcriptional noise, but those are not actually transcriptional noise. Those are really biologically functional transcripts because they are highly regulated, and specifically they are originating from the enhancer region. So these are called the enhancer. Active enhancer can give rise to this transcript that is called enhancer RNA. But how these enhancer RNAs are functioning, that was not known. So there are two previous reports. They are showing that like this enhancer RNA can interact with the like DRD4 or CBP. These are the histone modifiers. So this can interact with this DRD4 and CBP and can help their binding to the chromatin. So that's why I was interested to check that how this different type of enhancer can activate in different cancer cells. So I took the colon cancer cells, and there is a very common oncogene called MYC oncogene. And upstream of MYC, there is in a super enhancer region where from this HTK4 acetylation or HTK4 monomethylation deep-seq data, we have identified like there are different activation patterns. And from this activation pattern, we have identified these three are the enhancer region, and that is regulating the MYC oncogene. And there is another technique that is called GROW-seq. So from GROW-seq, this is the global run-on sequencing. From here also we can in vitro experiment, we can reduce the nascent transcript. And from this Rosic data, we found that this active enhancer is a mark of active enhancer like MYC490 here, and there is an active transcription going on. So there we can detect some transcript. So these transcripts that are originating from this MYC490 enhancer region, those are called ERNA, MYC-ERNA. So we call them MYC490 ERNA. And then we checked by real-time PCR that this MYC-ERNA level is very high in the cancer cells compared to the normal epithelial cells. And when I knock down this MYC490 kb RNA, they are knocked down and they can reduce the expression of MYC mRNA. That means these RNAs are very specific, and they are regulating the transcription of this MYC oncogene. Then we thought, okay, how this MYC-RNA is regulating the MYC mRNA expression. So for that, you need to know that what are the proteins those are interacting with this eRNA. But this MYC-RNA is very labile. Their half-life is very short. It's very hard to detect them, and it's very hard to work with them. So that was a challenge. And then there is a famous technique called RAPMS. So where we can use this biotolerated oligo to pull down this eRNA. So I standardized this technique from other labs, and then found that there's a biotolerated oligo-mediated pull down, and by this pull down there will be interaction of this eRNA with the protein, specific interaction, and this is very specific. And then after the mass spec analysis, we can detect what are the proteins those are interacting with this eRNA at a specific moment. So I took two different conditions like normal colon cancer cells and chronic infection colon cancer cells by TNF stimulation, and found that there are like dynamic interaction of different proteins with this MYC-RNA. For 0 hour there are interaction of different proteins, and for 16 hour there are different proteins. And I found ITS2 is a very interesting protein for myself, like is the histone modifier, and that is also interacting with MYC-RNA. And by UV experiment, I confirmed that okay, ITS2 is interacting with these two different RNAs. That's for sure. Then I looked why this MYC-RNA is interacting, whether they are interact, because there are other proteins those have the ITS domain. Like ITS2 is a protein that has a ITS domain. So its domain is specific for binding to the acetylated histone or protonated histone. But there are other proteins like AX9 or ENL that also have the ITS domain. But from this RNA MSTA, I found that like only the ITS2 protein is binding specifically to MYC-RNA. So when we add MYC-RNA to the ITS domain of its protein, there is a shift, means the MYC-RNA is binding to the ITS domain. Whereas for ENL protein, the ITS domain is not binding to the MYC-RNA. For AF9, I can see some binding, but not that much like ITS2 protein. So this indicates that it is very specific to binding to this MYC-RNA, 490 kb MYC-RNA. And from the in vitro, this histone octamer binding assay, I confirmed that this MYC-RNA is increasing the binding propensity of its domain to the protonated histone. So that means this MYC-RNA are helping its protein to bind to the specific region of histone protein or the chromatin, so that we can induce the transcription. And then to have some biological, cell biological context, I did the TIP-seq analysis from this TNF stimulated cells. And with TNF stimulation, I found there are a lot of histone proteination increases. So there is a two type of signal, histone acetylation and also histone proteination. Although the acetylation signal is the more compared to protein signal, but still the protein signal can be there in specific region of the genome. And this protein signal can be found by, and this ITS2 protein is a part of ATAC complex. So this ATAC complex, so ITS2 helps this ATAC complex to recruit to this protonated histone. It is evident from the genome-wide cut. And then I confirmed this with the MYC-RNA context and found that this ATAC complex are mostly recruited to this enhancer region of this MYC oncogene. So this indicates that maybe the MYC-RNA that is guiding this ITS2 containing ATAC complex to bind to the MYC enhancer region or the MYC promoter region to induce the MYC transcription at that situation. And so then to confirm that, I purified this eta complex from the nuclear extract. This is the eta complex, and use this purified eta complex to perform the in vitro chip. So here for the in vitro chip, I have used the chromatinized tablet, and all these things happen in vitro in test tube, and found that in presence of mic-RNA, there is a particular stoichiometric ratio. At this ratio, the binding is very high. That means in a particular this nanomolar ratio, each protein can have a high propensity towards the chlorinated histone. And when it is binding, the eta complex can acetylate the subsequent histone residue, the H4K16. And also found that not only the binding is high, at that particular stoichiometric ratio, the eta complex can induce the acetylation of H4K16. So from this, we conclude that like mic-RNA augments the binding of this H2 protein to protonated histone, and in this way recruit the H2 containing eta complex to a specific enhancer or promoter region to induce the transcription of the specific gene in chronic CNS stimulated cells. So it's almost there. So we are hoping to submit the manuscript soon. And it's very interesting that this eRNA, this non-coding RNA is not known that much. So they may have many different functions in different cancer cells or in different disease condition also. So that in this way, that would be very interesting to explore. So that way, I like to thank my supervisors, Dr. Subhendranath Bhattacharya, Professor Gauri Shankar Ghosh, Professor Alex Hoffman, and Dr. Shannon Lawbert for guiding me throughout all the way, and also the funding agency. And here at NIBMG, I have recently joined like 2 months ago maybe, and I'm very excited to start my new lab where I'll be working on the cancer epigenomics, mostly like in pancreatic cancer. So how this, when the cancer grows, it can create a tumor microenvironment, and how this different epigenetic modification can induce this tumor microenvironment or can regulate this tumor microenvironment. That will be one interesting area of research in my lab. Also, I'm very much interested to check this enhancer RNA that I last described. So how this enhancer RNA, whether there is any enhancer RNA production in the pancreatic cancer, and how they are regulating this tumor microenvironment or the cancer progression, will also be a very interesting part of my research. So in this way, I like to thank all of you for giving me the opportunity to work here and present my work here. Thank you. Thank you.
Thank you, Dr. Anup. Next up, we have a story of identification of Toxoplasma virulence factor 2 mechanism of parasite clearance by Dr. Debanjan Mukhopadhyay, who is also an NIBMG fellow here.
A very good afternoon to everyone. I would like to very much express my thanks to Professor Soumitra Das, Director NIBMG, and the organizing committee to giving me the opportunity to present my research work in front of all of you. So I recently joined NIBMG a couple of months back. So today I'm going to present one of my research work from my postdoctoral research, and we published this story already. So it's a very new, and to me it's a very fascinating story to start with a virulence factor, and then we identified how our immune system can sense this factor, and ultimately that factor helps the parasite to clear from our body. So before going into the research work, I would like to say a little bit of my background. So I did my PhD from Institute of Postgraduate Medical Research and Education, which is commonly called as IPGM here, and maybe all of you know as PG Hospital. So I did my PhD under the supervision of Professor Mitali Chatterjee, and I did my PhD work on translational immunology on patients with post-Kala-Azar dermal leishmaniasis, where I worked with mostly how the immune system has been dysregulated in patients with PKDL and how different immunomodulatory drug which has been given to the patient can actually help to get rid of the parasites and the patients benefit from the treatment. So from there, I moved to my postdoc to University of California, Davis, where I start my research work with another parasite called Toxoplasma gondii. And today I'm going to talk about that work. So Toxoplasma gondii is a very widespread parasite, and most importantly, Toxoplasma can infect almost all warm-blooded animals and all nucleated cells, including what the name, the cell type you can name, and it can infect anything except RBC, which is enucleated cell. So that's one of the fascinating part of this parasite. So the name Toxoplasma gondii, as you can see from this picture, from the left, the Toxoplasma, it's the Greek called bow-shaped, or it's kind of a like a partial moon-shaped structure. This is the electron microscopic structure of Toxoplasma, and the name gondii comes from this cute rodent called gundi. And the Toxoplasma actually is identified from this rodent almost 110 years back. So why we study Toxoplasma? So Toxoplasma is an obligate intracellular protozoan parasite, which means that Toxoplasma can live only within a host cell. So unlike Leishmania, which you can grow in vitro in a test tube or in a 15 ml Falcon tube, Toxoplasma you cannot grow like that. So you need a host cell into which if you put Toxoplasma, it will definitely infect, and then it will form a parasitovirus vacuole, which is here demarcated as like a star marks in yellow color. And in the fluorescent micrograph, you can see the green boundary, which is the parasitovirus vacuole and parasitovirus vacuole membrane. So inside that, Toxoplasma grows and then they multiply. So one of the disadvantages of being intracellular is that anything that forms a vacuole inside of a host cell can be targeted by the lysosome, but Toxoplasma has its own mechanism so that it can actually reside as a non-fusogenic parasitovirus vacuole membrane. And most importantly, why people have studied Toxoplasma? Because the phylum Epicomplexa, in which Toxoplasma belongs, includes another more medically relevant parasites. One is Plasmodium, which we all know causes malaria, and another one is called Cryptosporidium, which causes the infantile diarrhea. So both Plasmodium and Cryptosporidium, until few years back, were not able to be cultured, and they are not actually genetically amenable. That means if we have to study the parasite on the molecular side of the parasite, we need to do genetic knockout of the parasite virulence factors and those things, and those were not able to do by the Plasmodium and Cryptosporidium mechanism because they are very much strict in their genetic structure. So Toxoplasma has been used widely for that one, because Toxoplasma has some proteins which are very much similar in terms of amino acid and genetic sequences with Plasmodium and Cryptosporidium. So what Toxoplasma actually causes? So it actually prevalence very much in human, and almost 30% of human population of the entire world is already infected. That means if I can assume we are sitting 6 people in this room, and out of 6, 2 are already infected with Toxoplasma. But we rarely see someone is actually died or become very sick with Toxoplasma. Why? Because usually our immune system is strong, and that can prevent the growth of Toxoplasma in a superior way. But Toxoplasma is an opportunistic pathogen like many others. So it can cause disease, but when our health is somehow immunosuppressed, for example, when we have cancer, when we have any kind of organ transplantation like heart transplantation, liver transplantation, or when our mothers become pregnant, and that time Toxoplasma can cause its severe effect on us. For example, in the pregnant woman, it causes even abortion and preterm birth, which is a very important thing. Even for us, like we have the Garvini program running in India, and I think because in India it's kind of understudied, Toxoplasma, whether that can be another causative agent for this preterm birth and abortion, it can be looked further. So it also causes blindness, but these things are all strain dependent. So blindness is caused only by certain strains of Toxoplasma, which are mainly predominant in South America, mainly in Brazil, where almost 80% of the population get infected. Now the question is why Brazil or Western countries are so heavily infected? Because of the food habit. So toxoplasma comes into our body when we have undercooked meat. For example, in Western countries, they have like tartar, which is a fermented cured meat. So toxoplasma cyst can be present there and people can be infected. Like in France, almost 90% of the people are infected with toxoplasma only because they have this meat, they have their delicacy, it's called steak. So they like the steak, which is most of the time undercooked, and they also like this tartar, which is basically raw meat. But in Indian scenario is completely different. Everything we do overcooked actually. So for us, it's not coming through the meat. We most of the time get this parasite from the water, where it comes like in our case, water we can get the cyst or the oocyst from the toxoplasma and we get infected. So then the toxoplasma, how it manipulates our cells. So toxoplasma, like two other epicomplexan parasite, Plasmodium and Cryptosporidium, has three secretory organelles named as micronyms, which you can see like here, the blue color, the triangle shaped proteins. This is the micronyms, and then we have rhoptries, which are present as a rhoptry bulbs. It's a green in color in both the cartoon as well as the electron microscope picture. And then the other one is called dense granules. So what happens when toxoplasma invades into a cell? It helps through the micronyms, and then it can enter into the cell. And during its entrance, it secretes the rhoptries, which helps not only for its invasion, but also it manipulates the host cell signaling, and some of the rhoptry proteins can even enter into the nucleus. Now, once it enters, it then starts forming the boundary before around it, which is called parasitoporous vacuole, and it's the membrane bound, double layered membrane. So in this membrane, they are now starting the other important proteins called GRAS, which are secreted from dense granules, which is red in color. So these GRAS are also, they can manipulate the host signaling. They can be present in the cytosol, or some of the GRAS can go even into the nucleus to modulate the host signaling. So in this way, toxoplasma actually multiplies inside the cell, and when the multiplication rate is too much, the cell cannot take no more load, it certainly starts lyses out the cells, and then it enters out of the cells actually. It's called egress, and then it reinvades to the other cells. So this is basically the lytic cycle of the toxoplasma. But most of the interesting part is here, like how the ROPS and GRAS, they are actually modulating the host cell and the actually evasion of the host immune response. So how immune system reacts against toxoplasma? So far from long time, because toxoplasma is very much abundant in the Western countries, people have studied, but it always studied in the murine model. So recently we published a paper where we have shown that how toxoplasma actually manipulates human immune response. So basically what we have shown there is that when monocytes are infected with toxoplasma, then these monocytes have two things. Number one, they start secreting IL-12, which is a pro-inflammatory cytokines, and acts with other two important inflammatory cytokines, IL-1 beta and IL-18, which is secreted through the inflammasome activation. And then these trio of cytokines then act on the other immune cells, mostly if you do the cold culture like CD8 T cells and NK cells, then these cells become activated by the secretion of these cytokines, and they start secreting IFN gamma. Now, this IFN gamma is actually detrimental for the parasite because it can kill the parasite. So one of the question will be why parasite will suddenly stimulate something that can actually is detrimental for themselves. But this is another clever strategy by toxoplasma, because toxoplasma always needs to be inside of a host cell. So if it grows too much, then it will lyse the host cell, so it cannot get another host cell to grow. So it will keep itself in a shape that it can enjoy the host body, which is our human body, unfortunately, so that it can stay there forever. And it's a very important message that once we get infected, we cannot get any sterile cure for toxoplasma. That means no matter whatever treatment we can do or we can have so far, none of them can completely eliminate toxoplasma. So in that cases, actually toxoplasma is very dangerous. So we also treat the cells, especially the human PBMCs, with IFN gamma, and you can see from here that without IFN gamma, if we put the relative growth at 100%, with IFN gamma within 24 hours, the parasite growth becomes 50%. That means IFN gamma can kill the parasite. So what is the mechanism of parasite killing? Now, before going into the details of mechanism of the parasite killing, we need to know the other features and how immune system reacts at least in the murine cells, against toxoplasma. So if you treat any murine cells with IFN gamma, it will bind to its receptor, and then it starts the signal transduction pathway to the STAT1, and which upregulates many enzymes like NADPH oxidase that produces reactive oxygen species, then inducible nitric oxide synthase or that actually produces nitric oxide. And also IFN gamma in the murine cells have two important proteins called IRGs, which is called immunity related GTPases, and GBPs, which are guanylate binding proteins. So these two proteins are actually more active on toxoplasma. So what these proteins actually do? These proteins form multimer like the membrane attack complex during complement activation in our immune system, and these multimers of IRGs and GBPs then bound to the parasitoporous vacuole membrane, and then it just perforates the membrane, so that now the cytoplasmic content of the host cell, which contains many proteolytic enzymes and other stuff, they can have easy access to the parasite membrane, and in that case parasite become live. So parasites are also very clever. Some of the parasites, they are very resistant towards this IRG and GBP mediated mechanism. How? Because they contain certain proteins. For example, ROP18, which is a kinase, ROP5, which is a pseudokinase, ROP17, which is a transporter as well as a pseudokinase, and GRA7. This tetrameric complex actually keeps these IRGs and GBPs away from the parasitoporous vacuole membrane. And how? Because ROP18 is a kinase, they actually phosphorylate these IRGs, and once they become phosphorylated, they no longer can form the multimer, and therefore parasite can grow fine. So in that occasion, we can say type 1 parasites are resistant. But there are two other predominant types of parasites, type 2 and type 3. And type 2, they have this ROP5 as inactive form. So although they have ROP18 and ROP17 and GRA7 active, but because the ROP5 is inactive, then it no longer can phosphorylate these IRGs, and then IRGs can form these multimeric complexes on the PVM, and eventually the parasite will lyse down. And so they are called susceptible parasites. Same thing happens with type 3, but instead of ROP5, type 3 parasites have a polymorphism in ROP18. And it is important to mention that both ROP5 and ROP18 in type 2 and type 3 have basically a single amino acid change from what we have in the type 1, that is ROP5 and ROP18. So a single amino acid change in the protein structure can cause the resistant parasite to become susceptible parasite. So now the questions that we actually asked ourselves before we start our work, number one, is the stain-dependent susceptibility that we have seen in murine cells are also exist in human cells or not? Then what parasitic factors, or what parasitic factor, if it is one, governs this resistance and susceptibility against human IFN gamma stimulated cells? And what is the mechanism of this parasite killing in IFN gamma stimulated primary cells? Because in human, we don't have IRGs and GBPs. So in murine cells, they have IRGs and GBPs, but in human, we only have GBPs, whereas IRGs are not present. So these are the three questions that I will show you today how we address. So we first check the parasite strain-dependent susceptibility present or not. So we took two defined parasites, type 1, which is RH, and type 2, which is PRU. So in a 24-hour growth pattern, you can see that with IFN gamma, only 20% of the RH parasites are killed or eliminated from the cells, whereas type 2, almost 50% of the parasites become eliminated from the host cell. Whereas if we grow the parasites for more longer time inside the host cell, like for example for 6 days, you can see even more dramatic response. For example, we did plaque assay, like the similar like people do for viral plaque assay. So in plaque assay, you can see the plaque number after 6 days compared to the type 1 parasite. Type 2 parasite have much more loss, and same thing happens to the plaque area. So we can say that type 2 parasites are more susceptible than type 1 in IFN gamma stimulated human primary cells. So now the question is, what factors make type 2 more susceptible? But there is another question, that what factors make type 1 resistant? So today I will focus only on the type 2 factors that make them susceptible. Type 1 factor, we also did that. That was another story. We did a whole genome CRISPR-Cas9 screening to find out the factors that make type 1 parasite more resistant. So now what makes type 2 parasite susceptible to IFN gamma stimulated cells? So if we go deep into the how parasite secretes into the protein, as I mentioned that ROPTs and the dense granules are the proteins which are secreted into the host cell as well as on the parasitoporous vacuole membrane. So how it's secreted into the host cell and nucleus? So there is a big translocon called Mir translocon, which actually comprises 4 major proteins, Mir1, Mir2, Mir3 and Mir4, of which Mir1 is the predominant one and actually has the major effect. So we thought, okay, parasite actually secretes more than 20 types of proteins into the host cytosol and nucleus. Before checking each of them by making 20 or 22 knockouts, we can just make a one knockout of Mir1 and see if Mir1 knockouts become more susceptible or not. Because if we see Mir1 knockouts are more susceptible, then we can go one by one for the other factors. So we did CRISPR-Cas9 knockout of Mir1 from the parasite. And you can see when we knocked out Mir1, actually there is no difference in the susceptibility of the type 2 parasite. So compared to the wild type, both delta Mir1 parasites have equal number of plaque number loss as well as plaque area loss. So then we think there must be something else in the type 2 parasite. So we thought how to find the factor. So we choose 2 criteria. The major one is the protein has to be PVM localized protein. Why? Now, because PVM is the interface where the parasite protein can interact with the host protein, because some of the protein has its face in outside of the parasite and the face is close to the host cytosol, where it has more chances to get interact with the host cell. So the rationale was PVM localized proteins have more chances of interaction with the host protein. And the other criteria that we put is the protein has to be polymorphic. That means it is functional in type 2 but not in type 1. So we did Toxoplasma genomic database search, and then we found from QTL studies also that there is a couple of factors of them, but one of them is called Gra15, which is a highly polymorphic protein between different types of Toxoplasma. And most importantly, in type 1 Rh, there is no functional protein, that is, there is no Gra15. There is a huge truncation in deletion of the gene from the Gra15 in the type 1, but in type 2 it is highly active. So you can see from this immunofluorescence picture that an infected cell with type 1 has no NF-kappa B, which is actually red here, and in the type 2 infected cells, NF-kappa B is clearly in the nucleus. Whereas if we genetically engineer the type 1 parasite with the expressing the type 2 Gra15, you can see the NF-kappa B is in the nucleus. And when we delete the Gra15 from the type 2 background using the CRISPR-Cas9 system, you can see there is no NF-kappa B in the nucleus. So Gra15, the major function is NF-kappa B. So this story we also have published in a separate paper. So then we check whether Gra15 is really mediating this susceptibility of parasite or not. So what we did, we took the Gra15 knockout parasite that we make, and then we also using CRISPR-Cas9, we put back the wild type Gra15 in another loci of the Toxoplasma to make it a complemented parasite. And you can see from this graph, wild type parasite, they are susceptible, like almost 50 to 60% after 24 hours is eliminated. Knockout parasites are very much resistant, almost like 90% of them are still present after 24 hours. And the complemented parasite, they are showing kind of intermediate susceptibility, like around 70% parasites are still there. So we also did the infection per field, and you can see that compared to the wild type, delta Gra15 are more resistant, and similarly Rh, which is a type 1 parasite, are resistant, but when we put the Gra15 into the Rh parasite, it makes them susceptible. So it means that Gra15 mediates the susceptibility of the type 2 strain. So now the question is, what is the mechanism of the parasite killing? So we then go for how it in other human cells, like human transformed cell lines, like in HeLa, and neuronal cells like microglial cells, people have shown that when IFN gamma stimulation happen to those cells, they act on the STAT1 phosphorylation, then IRF1, and then it induces IDO or indolamine 2,3 dioxygenase, which actually catabolizes L-tryptophan to L-kylinurin. Now, what happens if tryptophan is degraded to the kylinurin? The thing is, parasite is auxotroph for tryptophan. So if we can omit tryptophan from the media or into the cells, then parasite will not able to grow. And that is what people have shown for HeLa and neuronal cells. So we thought maybe it is going to be the same for us in human primary cells also. So first we check by a STAT1 inhibitor whether IFN gamma really works in this way. And you can see that when we add the STAT1 inhibitor fludarabine, you can see almost like 50 to 60% parasites are almost here eliminated. And when we put fludarabine, you can see that the growth of the parasite is restored. That means IFN gamma mediated signal transduction pathway is actually now blocked by using the inhibitor. So then we check, okay, we will put back tryptophan into the medium and say we will try to see whether parasite growth restoration is happening or not. And you can see that the percent of plaque number or plaque area with IFN gamma is almost 50%. That means 50% parasites are eliminated. But when we put back L-tryptophan, nothing happens. So tryptophan supplementation into the cells actually not able to restore the parasite growth. So this pathway, which has been shown to be worked on HeLa and other cells, is actually not working on the primary cell. So then we thought of what is available in literature. So we were searching and we find that in some of the cells like human umbilical cord endothelial cells, which is a type of primary cells also, there ubiquitination of the parasitovirus vacuole membrane and then downstream autophagy pathway also works. So then we thought, okay, that is also a primary cell. So what if we will try in our primary cells, which is a primary skin fibroblast? So we check the P62, which is a autophagy adapter protein, and you can see from this confocal images that P62 actually surrounds after IFN gamma into the parasitovirus vacuole membrane. And when we quantify the percent of P62 into the parasite membrane, you can see that when we add IFN gamma, type 2 parasites or 2 have much more P62 compared to the RH, which is the type 1 or 2 delta graph 15. And then we also check LC3B, which is the autophagy adapter. You can see that when in IFN gamma stimulated cells, the autophagy adapter LC3B is also nicely coated around the parasitovirus vacuole membrane. And when we quantify, we also get a very similar result. So all these things turns into endolysosomal fusion. So we check with the LAMP1, which is the lysosome endosome fusion marker, and you can see that because lysosome has an acidic pH, so whenever this endolysosomal fusion happens, the parasites will be degrading, right? So you can see the green parasites are already much smaller and the green portion is kind of degraded, and you can see the LAMP1 is still there. So these actually tells us that endolysosomal fusion happens after IFN gamma stimulation in a human primary fibroblast. We quantify this result, and to ensure that this is the mechanism by which parasite is killed inside of the IFN gamma stimulated fibroblast, we also use an inhibitor of endolysosomal fusion called baphyllomycin A1, and you can see that in the IFN gamma stimulated cells, whereas almost like 50% parasites are died, with treatment with baphyllomycin, the parasite growth is restored. So this tells us, okay, endolysosomal fusion in type 2 background makes the parasite susceptible. So now the question is, how Graph15 influences IFN gamma mediated killing of parasites? So because we thought, okay, Graph15 activates NF-kappa B. NF-kappa B is an inflammatory signaling molecule, so we get our hit. It is probably because of this. So we thought if we inhibit the NF-kappa B signaling using an inhibitor, we will easily restore the parasite growth inside IFN gamma stimulated cells. So we did the same, but we didn't able to restore the parasite growth. So that means the NF-kappa B activation by Graph15 is actually a different pathway compared to its susceptibility in the IFN gamma stimulated cells. So then we thought what we should do. So then with the powerful approaches of mass spec and then bioinformatic analysis, we found that actually Graph15 interacts TRAP molecules, but in a very specific way. For example, with TRAP6, it binds only to TRAP6 in the presence of IFN gamma. So this immuno precipitation experiment, you can see that we use Graph43 as a control protein. So in the non-IFN gamma condition, Graph15 cannot bind TRAP6. It binds to TRAP6 only in presence of IFN gamma, whereas the control protein Graph43, it cannot bind TRAP6. So we thought, okay, maybe TRAP6 is the key molecule then. So what next? So then we thought we will look for the sequence analysis of Graph15, and there we find that actually Graph15 has two TRAP2 binding sites, AAEE amino acid and then SQQE, and one TRAP6 binding site, which is this PIGNZ. So we thought with the help of site-directed mutagenesis technique, we will mute this site in the Graph15 and see if the susceptibility pattern in IFN gamma will change or not. So we make the knockout this mutant parasite with TRAP2 mutant and TRAP26 double mutant, and just to make sure that the proteins are expressed in a proper localization, we check the localization, and you can see that Gra15 is present in the membrane as well as in the parasitovirus vacuole in all wild type mutant and the double mutant. So then we go for the first immunoprecipitation, that actually now it is not able to bind. So you can see that without IFN gamma, Gra15 binds to TREF2 but not to TREF6, and in the mutant of TREF2 or TREF266, the TREF2 binding is no longer present. So we were able to make the mutant, and with IFN gamma you can see that the TREF2 is still there in the wild type, and TREF6 it can bind. But surprisingly, we saw that in the TREF2 mutant, the TREF6 binding is actually much weaker. So that means TREF2 somehow helps recruitment of TREF6 also. So we make our decision by using this assay, that whether in IFN gamma stimulated cells compared to the wild type, TREF2 mutant or TREF26 double mutants are more resistant or not. And you can see from this graph, compared to the wild type, both the mutants are more resistant compared to the this wild type. So that means TREF2 and TREF6 binding makes the parasite more susceptible. So if we make a mutant, then parasite become resistant. So we also confirm this finding by able to make a knockdown cells from TREF6 from the cells. So we use scrambled and specific siRNA technique into the primary fibroblast, and we able to knock down the TREF6, and then we check the parasite growth, and you can see compared to the scrambled, which is a control where parasites were 50% killed after 24 hours, in the TREF6 knockdown cells, the parasites were not able to kill the inside the host cell. So that is the model that what we have found, and we published this paper also. So IFN gamma stimulates the Gra15 mediated TREF6 recruitment, which recruits P62 and then LC3B GABARAP, and then it causes the endolysosomal fusion. So with this, I would like to finish my talk here. I would like to acknowledge Professor Jeroen Sai for giving me the opportunity in the postdoc in his lab, and also all our members, which you can see, this is our cell culture lab. This is one of the busy day in the lab where 6 people are working in 4 hoods, and this is our big group. I would like to thank UC Davis DNA Technologies and Genomics Court, American Heart Association for providing me 2 years of fellowship as well as a grant, and NIH for overall grant. Thank you. And so my new lab at NIBMG will focus on 2 most important projects that I started at during my PhD, both of them with Leishmania. So project 1 will be the genetic and molecular basis of selective cellular tropism of intracellular pathogen. We will use Leishmania as a model, how cell autonomous innate immunity will be used to kill the pathogen inside the cell. So here we will use RNA-seq and other methods to establish why Leishmania is only infect on the macrophages but not the fibroblast or keratinocytes. And in a project 2, which will be more directed to the translational, that is with patient samples, where we will assess the genetic association of vitamin D metabolism and its functional role in immune response against post-Kala-Azar dermal leishmaniasis. So with that, again, thank you. I would again like to thank our director and the organizing committee to giving me the opportunity to present my work.
Thank you, Dr. Devanjan. I am sure with this we come to an end of the talks by our young faculty, and I am sure our audience, students as well as the scientific fraternity would have loved to interact. Unfortunately, these are unprecedented times and this is the new normal. So science is also on a monologue right now. Moving on, we have 2 more talks by our senior PhD students. First up, we have Ms. Devashri Tagore, and she will be taking us through an interesting talk on the genetic history of India's earliest settlers. Over to you, Devashri.
Good evening, everyone. Am I audible?
Yes, you are audible.
Yes, today I'll be talking about the genetic history of India's earliest settlers. Now, it is a well-established fact that the birth of modern humans was in Africa. So whether it is from finding deep mitochondrial lineages within the African subcontinent, or discovering early modern human fossils across Africa, or finding highest phonemic diversity within the African subcontinent, all lines of evidence, whether it's genetics or archaeology or linguistics, points to the fact that modern humans originated in Africa. And this happened approximately 200,000 years ago. But it wasn't until 60,000 years ago that the first out of Africa migration, that is the first successful modern human migration out of Africa, happened, which eventually led to the peopling of the entire planet. One of the earliest places where the out of Africa migration brought people was India and Southeast Asia. This region occupies about 5% of the total landmass of the world, but harbors over 26% of the world population. And this population is genetically, linguistically, and culturally diverse. Of the different populations residing in this region, the Austroasiatic speakers are one of them. And you can see in the map the distribution of these Austroasiatic speakers, and this is sort of a scattered distribution. Previous multiple studies, including some by Professor Majumdar and Dr. Basu, has found that the oldest mitochondrial and Y chromosome lineages are present in the Austroasiatic speakers of India than in any other population of India, which points to the fact that the Austroasiatics of India are the earliest settlers of this region. Similarly, the Austroasiatics of Malaysia have also been found to be the earliest settlers of peninsular Malaysia. There is a conundrum about the Austroasiatic presence and their movement. One hypothesis suggests that the Austroasiatics were first in India and then moved to Southeast Asia, while the other theory suggests that they were first in Southeast Asia and then moved westwards to India. Another theory suggests that the Austroasiatics were in southern China and eventually moved to India and Southeast Asia. There is another theory which says that the spread of Austroasiatic language is associated with the spread of rice cultivating farmers. But if that were true, why do most of the present day Austroasiatics are mostly hunter-gatherers or primitive agriculturalists? So to resolve this conundrum, we did this study and we obtained the genomes of individuals from different populations, as you can see in the map. This data set includes individuals from populations of India belonging to 5 major genetic ancestries, as was inferred by Professor Dr. Basu et al. In 2016, and these genetic ancestries are ancestral North Indian, ancestral South Indian, Tibeto-Burmans, Austroasiatics, and the island ancestry. Along with that, we also obtained genomes of 2 linguistic groups from Malaysia, the Austroasiatics and the Austronesians. Now, I would urge everyone to keep a note of these abbreviations, because in the upcoming slides, I'll be using them very frequently. The rest of the individuals were obtained from the Human Genome Diversity Project. The Austroasiatic language family has over 150 languages, and these languages are broadly classified by linguists into 3 major language groups. The Munda language family, which comprises of languages spoken by the Austroasiatics of Central India, the Khasi Khmer subgroup, which is a language spoken by the Khasi tribes of Northeast India, and Mon Khmer, which contains the languages spoken by the Austroasiatic populations of Southeast Asia. And for our study, we obtained genomes of 5 populations from the Munda subgroup and 6 populations from Malaysia belonging to the Mon Khmer subgroup. We began by doing a population clustering analysis, which is called an admixture analysis. So what happens in admixture, that given a population data set, one tries to identify the maximum or the optimal number of genetic ancestries that best define the population set. So for example, here we have all the Indian and the Malaysian populations and a few populations from their GDP data set, including the Central South Asians and East Asians. And there are 9 distinct genetic components that best define the data set present here. Each vertical line that you see is an individual's genome, and that is colored based on the proportion of inferred ancestry. So this individual has 3 inferred ancestries from the 9 ancestries that are inferred for the entire data set. So this is how you interpret the admixture graph. So what we found was the Indian and the Malaysian Austroasiatics have distinct genomic composition, that is Indian Austroasiatics of purple and the Malaysian Austroasiatics of largely yellow, and the genomic component which is model to one Austroasiatic is absent from the other. So despite this linguistic commonality, we find that they are genetically quite distinct. Language has often been considered a good predictor of genomic differences, especially in context of India. Therefore, keeping that notion in mind, began by doing another population clustering analysis. So we did a principal components analysis on the individuals that we had in our data set, which comprises of the Indian and the Malaysian populations. So what PCA does is that if the populations which are genetically very similar tend to form distinct clusters which are close to each other. And what we found was that the Indian and the Malaysian Austroasiatics, despite the linguistic commonality, formed clusters which are far away from each other. However, there was a third population, which are the Tibeto-Burmans of Northeast India, who sort of formed a bridge between these two Austroasiatic groups. And in fact, in our phylogenetic study as well, we found that while the Austroasiatics weren't clustering together, the Tibeto-Burman population actually were clustering with the Malaysian Austroasiatics. So there is a linguistic genetic discordance that we find here, because the Tibeto-Burman speakers speak language which is very different from and does not even belong to the Austroasiatic language family. So here the Indian Austroasiatics, the Tibeto-Burmans and the Malaysian Austroasiatics. One of the factors that could have contributed to this genetic similarity between the Tibeto-Burmans and the Malaysian Austroasiatics is the fact that they could have shared a very recent common ancestor. In order to identify that, we looked into segments which are called identity by descent. So these are identical DNA segments that are inherited from common ancestor. As you can see here, the blue colored segment between these two individuals is IBD because it has come from this common ancestor. So if we find IBD segments between unrelated populations, then we can speculate that these populations had common ancestor. The longer the IBD segment, the more recent the common ancestors are. The more recent the common ancestor. So what we did was we looked into the distribution of these IBD segments between the two Austroasiatics group and between the Austroasiatics and the Tibeto-Burmans. What we found was that there was no significant difference in the IBD segment length. However, the mere presence of IBD segments indicated shared ancestry somewhere far back in time. So far, we have found evidence of shared ancestry between the Austroasiatics and the Tibeto-Burmans, but we still do not know why the two Austroasiatic groups aren't genetically the closest, or why the Malaysian Austroasiatics and the Tibeto-Burmans cluster together. So if you go back to the admixture plot, you will see that the Indian Austroasiatics have a green color component which is model to the Dravidian speakers or ASI of India, while the Malaysian Austroasiatics have a pink color component which is model to the East Asians and the Tibeto-Burmans. This is indicative of gene flow between these populations, that is local admixture of the Indian Austroasiatics with the ASI and of the Malaysian Austroasiatics with the Tibeto-Burman and the East Asians. We also did another statistical test which is called D-statistics, and we found that the East Asian populations had significant admixture with the Tibeto-Burmans, which is very well documented in past, but they also had significant admixture with the Malaysian Austroasiatics. However, the East Asians did not admix with the Indian Austroasiatics. Now it was time to look into these admixed segment lengths in the Tibeto-Burmans and the Malaysian Austroasiatics and estimate how long ago this admixture event took place. So we looked into East Asian ancestry segments in the Tibeto-Burmans and Malaysian Austroasiatics and found that these segments were large in the Tibeto-Burmans. From these segment lengths, we estimated the admixture date range to be between 22 to 6 generations ago, that is 500 to 140 years ago. We were also able to find the the time of split of these two Austroasiatic groups, that is when was the last time they shared common ancestor, and we found that the ancestors of present-day Indian and Malaysian Austroasiatics separated nearly 470 generations ago, which is 12,000 years ago. And this 12,000 years ago is important because this is a pre-Neolithic event. So the fact that the Austroasiatics language was brought by the East Asian farmers or was a result of the advent of agriculture that was earlier hypothesized does not hold true because Austroasiatics were already in the region before the advent of agriculture. We also looked into the change in effective population size, which in the Malaysian Austroasiatics showed a gradual decline, but the effective population size in some of the Indian Austroasiatics showed an increase, and after which there was a rapid decline. And this decline started around 350 generations ago, which is approximately 8,000 years ago. And this time actually coincides with the advent of agriculture, that is when the movement of the rice cultivating farmers from East Asia began. So the model that we proposed is that the Austroasiatic ancestors were widespread across Central India to Southeast Asia. 470 generations ago, the ancestors of present-day Indian and Malaysian Austroasiatics separated. And then around 350 generations ago, the migration of East Asian farmers began. Now, they did not enter India, but there was local admixture of Indian Austroasiatics with the ASI populations, while there was admixture between the Tibeto-Burmans and the East Asians and the Malaysian Austroasiatics till about 6 generations ago. Now I would like to switch gears and talk about something which I consider is a genetic time travel. So we are talking about ancient DNA. So inferring anything about the past from the genomes of present-day populations is often tricky because populations that we see are present at a particular geographical region at this point of time may not have been there hundreds of years ago. Populations are mobile, they keep moving, and while moving they admix with genetically distinct populations, and over time their genetic composition changes. So the ideal scenario to talk about distant past would be to obtain the genomes of individuals who were residing hundreds of thousands of years ago. And this is where ancient DNA has revolutionized the field of human genetics. So what happens in ancient DNA technology is that the archaeologists dig up bones and other fossil remains, and these are radiocarbon dated. So you know that when was the individual living in that region. These bones contain small amount of DNA. So a fragment of these bones is removed and it is grounded into a powder. This ground powder has the DNA. So the powder is dissolved in a solution so as to release the DNA in the solution, and that DNA is extracted and sequenced. So now it is possible to obtain the genome of individuals from bones and other remains who lived hundreds of thousands of years ago. So for our analysis, to substantiate about our findings and our model, along with the extant individuals that we have been talking about so far, we also pooled genomic data from 43 ancient individuals or archaic individuals from Southeast Asia and from 2 different studies. So along with the modern humans, we now have 43 archaic human genomes, and the oldest archaic genome in our data set is as old as 8,000 years, and the most recent is about 200 years old. So we have individuals from 8,000 years ago to about 200 years ago. And what we did was we did an admixture analysis with the Austroasiatic populations, the Tibeto-Burmans and East Asians, to infer the ancestral components. And on these inferred ancestral components, we projected the ancient genomes that we had. And what we found was that, so here we have the older archaic samples and here we have a newer archaic samples. And we found that there was a decrease in AAI-like ancestry as we moved from older samples to newer ones, and an increase in East Asian-like ancestry from older to newer. So to summarize, what we see is that the older archaic samples were more like present-day Indian Austroasiatics, because like the present-day Indian Austroasiatics, they had minimal East Asian ancestry, while the newer samples, due to East Asian migration and admixture, were more like the present-day East Asians and Malaysian Austroasiatics. So what we see is, over a period of 8,000 years, there is ancestry shift in Southeast Asia. So the key highlights of this study was we were able to validate the widespread distribution of Austroasiatic speakers across Central India and Southeast Asia who likely spoke the Proto-Austroasiatic language, whose remnants we see today in the Austroasiatic speakers of India and Malaysia. The ancestors of these 2 Austroasiatic groups separated long before the advent of agriculture, and though they locally admixed with linguistically and genetically distinct populations, their linguistic identity largely remained intact. And with the East Asian migration, we see ancestry shift in Southeast Asia. We recently published these findings in BMC Biology in March of this year. So for all this work, I would like to thank my supervisor, Dr. Annalabha Basu, for his support, and a bunch of other people, especially Professor Partha Majumdar, and our collaborators from Monash University, Malaysia, Professor Maud Phibbs, Dr. Farhang Aga Khanian, and Rakesh Naidu. My supportive lab seniors, my entire all my lab members, and special thanks to the director NIBMG and Professor Sharmila Sengupta, and the funding sources DST and DBT. Thank you.
Thank you, Debashri. Next up, we have Mr. Subhashish Ghosh. He'll be talking on deciphering the mechanism of plastic state of stemness in oral cancer.
Hello everyone, am I audible? Good afternoon everyone. First I would like to thank you, our director and organizer, for giving me this opportunity to share my research work. I am Subhashish Ghosh, DBT SRF, and I am working under supervision of Dr. Sandeep Singh. Today I am going to discuss the research work, and the title is Deciphering the Mechanism of Plastic State of Stemness in Oral Cancer.
Oral cancer ranks among the top 3 types of cancer in whole Southeast Asian subcontinent, and in India, oral cancer accounts for over 30% of all cancer. Gingivobuccal region of the mouth is the most common site for oral cancer occurrence, and that is quietly correlated with the habit of tobacco chewing among Indian population. So despite the improvement in the standard treatment strategies, the overall 5-year survival rate still remains around 50% since decades. The rate further drops for all patients who have this local regional metastasis and the lymph node spread. So oral gingivobuccal oral squamous cell carcinoma has been well catalogued, and its molecular nature and signature has been well documented by our ICC NIBMG project group. So going into further, so mutational profiling of this oral cancer patients has been demonstrated that there are 10 significantly and frequently mutated genes, and among them Notch-on is one of the most frequently genes. So this cluster demonstrates the magnitude of Notch-on receptor mutation, but when we see the mutational pattern for HNSCC, that is quite different from the Notch-on mutation pattern, which generally found in leukemia and other hematopoietic cancer as well as in breast cancer. So there in leukemia and breast cancer, the mutational Notch-on mutational type is like gain of function in nature, and that promotes constitutive Notch activity. But when it comes to HNSCC or head and neck squamous cell carcinoma, the extracellular domain of Notch got mutated, and in that way, it inactivates this Notch pathway in HNSCC patients. Coming into this, its significance in the patient survival, when we took this TCGA data sets and took this 4 Notch receptor mutational pattern, that shows that the patients in which the Notch receptors got mutated, they are showing poorer prognosis compared to the patients group who have active Notch. So this has showing its clinical significance. So to understand it deeper, then we took the help of further studies. So first thing is this, how this canonical Notch signaling pathway works. So canonical Notch signaling pathway works in a juxtacrine mode. That means that signaling interaction occurs between 2 neighboring cells. There are 4 Notch receptors, Notch-on 2, 3, 4 in case of mammals, and there are 5 Notch ligands, that is DLL-on 3, 4 and JAG-on and 2. When we took this help of this 3D spheroid culture and compared the gene expression pattern between adherent 2D culture and 3D spheroid culture, then it shows that along with other stemness markers that include SOX2, KLF4, ALDH1A, c-MYC, the Notch receptors, Notch-on, and the major downstream gene HES1 also got upregulated in stemness enriching 3D spheroid culture of SCC070, that is another OSCC cell line. So how it comes to stem cells, like stem cells model has been hypothesized that a few subset of cells, cancer cells, have the ability to stem-like property, and they can able to long-term regenerate itself with the property of self-renewal and differentiation, and therefore these cells are suggested to be responsible for cancer initiation, progression, metastasis, therapy resistance, as well as recurrence. So to study squamous cell, oral squamous cell carcinoma, like other solid carcinoma, and to delineate its or demonstrate its aggressive behavior, we adopted this stem cell culture model. Going into further, like to mimic this Notch inactivation in vitro culture model, we have adapted or employed this gamma secretase inhibitor. Gamma secretase is the enzyme responsible for cleavage of Notch intracellular domain, and by that way, it activates Notch pathway. So for pharmacological inhibition, we took help from these 2 gamma secretase inhibitor, LOI and RO, as well as transient gene knockdown has been done by using siRNA mediated knockdown. Both of these methods has shown its efficacy for downregulating Notch-related genes that include Notch-on, the receptor-on, and HES1 and HEON are the major 2 downstream genes of this Notch pathway. Then to demonstrate its functional role in in maintaining stemness, we plated the cells in 3D spheroid culture in presence of
in presence of this gamma secretase inhibitor LOI and RO at a 5 micromolar dose, and it shows that and it has shown that in presence of gamma secretase inhibitor there is upregulation for sphere formation efficiency, and it has been throughout these 3 different cell lines it is showing the similar pattern. So that demonstrates that Notch inactivity, though Notch pathway is involved in maintaining stemness, its inactivity does not abrogates that self-renewal property of stem-like cancer cells. In fact, it is showing higher efficiency. That means that Notch inactivity may lead to a different state of stem-like cancer cell in OSCC. To go into deeper, I have made this stable clone from OSCC cell lines based on this Notch constitutive Notch activity and inactivity. For that, I have transduced this OSCC cell lines with lentivirus mediated transduction, and I have generated 4 different clones. This P-lenti I kept as a control one. I have took the help of HS, that is the wild type HES1 overexpression, BHES is the mutated HES overexpression clone, as well as I have took HICN, that is HICN overexpression. That means activity of Notch pathway will be stably constitutively active in this transduced cell lines. Then I plated these cell lines for sphere formation, but before that I validated the cell lines and clones by doing QRT PCR, and QRT PCR result is showing that in Notch active clones there is a higher expression for Notch1 receptor as well as major downstream gene for both the OSCC cell line, that includes BC02 as well as SCC070. When it comes to sphere formation efficiency, it's going with the gamma secretase inhibitor results, and the pattern is similar, means that in absence of Notch pathway there is no abrogation for sphere formation efficiency. In fact, it is maintaining, and in some cases the efficiency is higher compared to the other three conditions. So it is further strongly showing the involvement of Notch pathway, and Notch pathway mediated 2 different states of stem-like cancer cells. The previous literature is showing the involvement for Notch pathway in maintaining this resistance against cisplatin and other platinum-based drugs. Then we took the similar approach, and we treated the stem-like cancer cells in 3D spheroid culture, but that shows that though there is cisplatin treatment, but there is basically there is enrichment for stemness markers that I have proven by doing QRT PCR, and it is showing that upregulation for SOX2, ALDH, ONA1, KLF4, Notch1, and HES1, the Notch genes. So from this previous preliminary data and reports, we have hypothesized that different Notch-related states of stem-like cancer cells may confer cisplatin resistance in oral stem-like cancer cells. Then also, then I have done this QRT PCR from this wild type HES and mutated HES clone, stable clone, and I'm showing that, and it is showing that Notch on and ALDH on A, that is that stemness factors, both are correlated in expression, means with Notch inactivity, both Notch on and ALDH on A is going down. So that means that Notch on activity is very much going with that other stemness marker, including ALDH1. Then what I have done this aldaflor assay. So aldaflor assay from this control and cisplatin treatment cell line, and the cisplatin treatment is showing the enrichment for ALDH positive cells, that is in the green, can compare. And it is in a similar fashion, it is also showing that Notch positive cells are going up in cisplatin treatment conditions. So then it is clearly showing that involvement of Notch pathway to maintaining this different state of stemness in stem-like cancer cells in 3D spheroid culture, as well as its role in cisplatin resistance. Then I have done this sphere formation assay and viability. I have checked this viability from these 3 different cell lines, where I have shown that in presence of cisplatin and Notch inactivators, there is lower sphere formation efficiency, as well as the viability has been significantly reduced throughout these 3 different OSCC cell lines.
Then I have done this MTT assay from Notch active HICN clone as well as from BHES, that is the mutated HES clones that depicts that Notch inactive state, and it's showing that in Notch inactive state of this stable clones there is significant reduction in the IC50 value for the cisplatin. Then this model comes from this results that due to cisplatin treatment, there is the low notch low cells of stem-like cancer cells can become notch high cells, and that can be targeted in cisplatin in presence of LY and RO. That's showing that notch is the notch mediated. There is notch mediated plasticity, and notch is basically induces this cisplatin resistance. So in presence of gamma secretase inhibitor, this is cisplatin can be more effective. To understand the biology behind this, we have done this RNA sequencing from the HES clone, BHES and HS, and you can see that there is in notch inactive clones, that is BHES, there is upregulation for JAK-STAT signaling pathway. The pathway enrichment analysis has been shown that in BHES clone, this few pathways has coming out, and JAK-STAT was one of the significantly upregulated pathway in absence of notch activity. Then we took this sphere formation efficiency, and as to consider that that is there co-inhibition of both notch1 and JAK1 could reduce the sphere formation efficiency as well as stemness. So we have used this tofacitinib and ruxolitinib. These two are JAK-STAT signaling pathway inhibitor, along with that LY and RO. Throughout these three different OSCC cell lines, the efficiency is showing that co-inhibition of both this notch and JAK is significantly reduced the sphere formation efficiency as well as viability. I have further checked this scenario in HICN and BHES clones. BHES clone is the notch inactive clones, and it is showing the similar pattern. That means that in presence of JAK-STAT signaling pathway inhibitor tofacitinib and ruxolitinib, there is significant reduction in sphere formation efficiency for both this GBC02 as well as SCC070 cell lines. So then this result has been further demonstrating that there is synthetic lethality is working. Notch is involved basically in mediating this plasticity between notch high and low, and in notch low condition, there is the JAK-STAT signaling pathway got upregulated. So we can inhibit this sphere formation by using notch pathway inhibitor LY and RO, along with the JAK-STAT signaling pathway inhibitor ruxolitinib and tofacitinib. In that way, the synthetic lethality is showing therapeutic promising results. So, along with, I am concluding my presentation. The conclusions are: notch inactivation is leading to a different state of stem-like cancer cell in oral squamous cell carcinoma. Conventional platinum-based chemotherapeutic agent cisplatin favors the enrichment of notch pathway that can be sensitized by using notch inhibition. Notch inactive state of stemness demonstrated its dependency on JAK-STAT signaling pathway, and we have demonstrated the synthetic lethal interaction between notch and JAK-STAT signaling in oral stem-like cancer cells. Thank you.
Thank you, Shubhashish. With that, we come to an end of our first scientific session of the program. We already have Dr. Shiv Pillai logged in. Welcome, sir. Right now, we'll be taking a short tea break, and we will resume back for the second session for the Foundation Day lecture by Dr. Pillai, sharp at 6:15. Thank you. Camera.
you will be able to calculate an odds ratio for delta.
Hi, Dr. Shiv.
Hello, how are you?
Hi, how are you?
I'm very good. How are you, Soumitra? Yeah.
You to unmute, or maybe you are just not. Yeah.
Am I muted?
No, so I can't hear you.
You can't hear me. Okay, let me just fix my sound. 1 second.
So probably they haven't connected you.
I don't know.
No, I can, sir. I can hear your voice. Both of you hear your voice.
Okay, but I don't know if will I be clear enough for everyone now or not?
Yeah.
Okay. Soumitra, maybe your speaker is low, or I don't know. I'm just trying to figure out.
Yeah.
So Bikram, you can hear me fine, right?
Yes.
সব কিছু শোনা যাচ্ছে তো, হ্যাঁ. Okay. Yeah.
Can't hear.
I'm wondering whether your audio is low. Bikram, I think Dr. Das's audio might have some.
Yeah. Let me check.
Bikram,
sir. হ্যাঁ স্যার, আপনি একবার ডিসকানেক্ট করে কানেক্ট করবেন একবার। স্যার আমি ওনাকে বললাম একবার ডিসকানেক্ট করে কানেক্ট করার জন্য।
হ্যাঁ ঠিক আছে। পুরো সেশনটা কি ভার্চুয়াল?
হ্যাঁ ভার্চুয়াল।
আচ্ছা। সো ব্রেক টাইমে কিছু খাওয়া দাওয়া পায়নি কেউ? হ্যাঁ, সিঙ্গারা ছিল না?
আছে স্যার, এবার শুনতে পাচ্ছেন?
মনে হচ্ছে
স্যার unmuted. Still you can't hear me.
We can hear you.
Oh, I can hear you now.
Okay.
All right.
We could hear you, but you were not able to hear us.
Right.
Yeah, I was just saying that if it's virtual, then no shingaras and all that today.
I know. So no. Actually, they are serving here shingara.
You didn't send me anything. আমি কিতে পাচ্ছি এখানে?
কিছু পাচ্ছেন না। So have you been to Kolkata at any time?
Kolkata is my hometown.
Really? Where?
I grew up in basically Bhawanipur.
Bhawanipur, I'm from
I was on Elgin Road. I lived on Elgin Road.
Yes. So actually, NIBMG has one, we have an annex, which is PG Polyclinic, upstairs of PG Polyclinic, which is close to Elgin Road.
I know that place. I was in IICB also, so I lived then in Baliganj.
I see. Oh, you are in IICB. Which year?
I was with Bachawat. I did my PhD with Bachawat.
I see.
So I was in Vellore with him as a medical student. Okay. And I did my PhD with him. I came with him from Vellore to IICB.
Oh, okay. So you might be knowing Devi Sarkar then.
I know Devi. Of course, I know Devi very well. Devi is on email with me once or twice a week.
I know. I see. So Anand Bachwat is also, I'm in close touch with Anandada. So,
Anand was a postdoc with me.
Oh, really? Achha.
He came to Boston. He was a postdoc with me right at the beginning when I started my lab.
Right. So I'm in touch because he's in ISAR Mohali.
Yeah, I know.
Right.
I was supposed to give a talk in March last year. I came to Delhi. I spoke at the institute where Gagandeep Kang was, right? At Faridabad. THSTI. I taught a course in THSTI. I was supposed to fly to Chandigarh to see Anand and give a talk, but unfortunately, COVID was a problem, so they cancelled. So I didn't go. Yeah.
I know. So, we are just waiting until this situation is better so that we can get you here and get you samosa and rasgulla, shingara.
what about kosha mangsho and dimer dalna and aloo posto?
Please come. I will treat you in my here at home here.
I eat over here also.
That's good. That's good to hear.
खूब भालो इलिश पाया जाए.
Right. अच्छा. That's nice. I know. Actually, I was in Los Angeles. I know. So I was in Los Angeles for 7 years, more than 7. So we used to go to some Bangladeshi shop and used to get really almost like 2 kg ilsha. It was very good.
Poddar ilish comes.
Poddar ilish, exactly. So LA is famous for all Bangladeshi shops.
That's true.
Yeah. So I'm sure Boston will is a lot of Bengalis, so you'll get Bengali.
Plenty.
Right. So
even Bengali restaurants are here. Good Bengali restaurant. Right. West Bengali restaurant also. Very good.
That's nice. Because I'm actually now in IISc for now last 23 years. In Bangalore, we don't have that many shops, so you don't get good fish.
একটা ও ক্যালকাটা আছে ওখানে একটা রেস্টুরেন্ট
ও ক্যালকাটা আছে কিন্তু ওখানে ইলিশ মাছ খেলে you don't feel like that is ilish. It looks like you are having salmon.
Yeah. It's not perfect. Yeah.
Right. You have to have O Calcutta in Calcutta. I had it. That is good.
Well, when I was in Kolkata, there was very few Bengali restaurants. When I was a kid, I went to high school in Calcutta. But at that time, there were no Bengali restaurants. There was one Bengali restaurant, it was called Suruchi. That's it. I mean, you could get Anadi cabin, Mughlai paratha and all that, but no real Bengali food. You had to eat in the house only. But now there are some restaurants in Kolkata.
See, traditionally, Bengali used to invite people and cook at home. So this outside...
Yeah, I know. Outside, they would eat Mughlai paratha.
Right. So even now I can see, compared to Bangalore, in my Calcutta home, they are not so fond of going outside and eating outside food. But in Bangalore, my daughter keeps doing that.
No. In Calcutta, they used to go outside, but not to Bengali restaurant.
They will go for dosa.
Dosa, other things, but not to Bengali.
Right. It's very nice to... So how long you have been there now? Almost. So you know Ashim Dasgupta, you have overlap.
Ashim also trained in the Baltimore lab before my time.
Before you,
because I've met him.
In 86, 88. I was thinking Ashim Dasgupta might be.
He was not in the Baltimore lab between 83 and 88.
He probably left in 82 and joined UCLA.
Exactly. So I didn't overlap with him. So the Bengalis I overlapped was Ranjan Sen and Shankar Ghosh.
Shankar Ghosh also you overlapped? I see. Okay.
Shankar and I are very good friends. Ranjan and I are very good friends. So,
Right. So I worked with Ashim Dasgupta with polio virus and then we worked with hepatitis C.
So then you're also connected to the Baltimore family.
Yeah. Steve Smale, Mike Carey, they were all in UCLA.
Steve is a good friend.
Yeah.
Very good friend. Yeah. Steve and I overlapped.
Steve and you overlapped. I see. Right. Yeah. I think when I was in UCLA, Steve joined.
Yeah. He would have joined about 89 or so.
Yeah. I joined in 1991. Yeah. Around that time.
Around then.
Right. Mike Carey was there for a long time. Mike Carey, I don't know whether he's still continuing there. So, we used to call Baltimore Tree. So you are one of the branches of Baltimore Tree.
That's right. I still see him once a year now, at least. But he's on our scientific advisory board here. So
I see. So is he still continuing in Caltech?
He's closing his lab down. It's almost closed now. He's 82 now, but it's not. He's slowing down a bit. Yeah.
He came to IISc, I think, a couple of years back. Last time I saw him, he visited our department. We had a long chat. Came to lab also, and then I work on hepatitis C. So I described the work. He's pretty alert.
No. He's very knowledgeable and on virology and immunology is very strong. No.
Right.
Is So, Soumitra, I have to be at the next meeting.
I know. So I think with just 1 minute, I think we'll start.
Yeah, that's fine. Just letting you know that I will have to. Yeah.
No, we're just in 2 minutes, we'll start. Bikram, just make it online in another 2 minutes.
Yeah, that's fine.
Yeah.
The working hours seem very different from IICB. Bikram told me you all are there till 6:30 in the evening, which is unusual. IICB people went home early.
See, Calcutta people, yeah, because they have to commute. They usually go by other places, and who are resident here. Now I think there are 3 faculty who
Good evening, everyone. Welcome, Dr. Pillai, and welcome to we'll welcome back our audience, the online audience, for our second session, which is also a Foundation Day lecture. I'd like to invite Professor Das to please introduce our speaker for today.
So, welcome, Dr. Pillai. Actually, it's a great pleasure and honor, privilege to introduce you. Thank you for agreeing in a very short notice. I know how busy you are, but it's very. I'm very happy to know that you had your roots in Calcutta and you enjoyed the Bengali food, and which most of these people who are celebrating our Foundation Day today had a probably have it. We missed you, but next time I think we'll make sure that you physically present here, and then you enjoy the snacks along with your talk. So it's, as I told you, it's a real privilege and honor for us that Professor Pillai is with us today for the Foundation Day lecture. He is the Professor of Medicine and Health Science and Technology at the Harvard Medical School and the Reagan Institute Investigator. He is the Program Director of the NIH-funded Autoimmune Center of Excellence at the Massachusetts General Hospital and the Director of the Masters of the Medical Science in Immunology program at the Harvard Medical School. So Dr. Pillai was just sharing with me, actually he actually graduated from Kolkata, then moved and completed medical studies at CMC Vellore, and that's in 76, way back, and subsequently obtained his degree in IICB Kolkata and continued his postdoctoral training in David Baltimore's lab. And I was just talking to him. There are so many renowned scientists like him, which we call Baltimore tree, and we just respect them for their science. And it was in David's lab, Dr. Shiv has actually shown the importance of the surrogate light chains for the B cell development, which actually binds to the antibody heavy chain in the pre-B cells. That's his discovery, and he's continuing in that. And in '88, he joined the faculty of this Massachusetts General Hospital and the Harvard Medical School. His laboratory at the MGH postulated the provided evidence for the first ligand-independent signaling model during lymphocyte development, which is now, it's a widely accepted mechanism for both pre-B cell receptors and the pre-T receptor signaling. And his lab continues to study and the basis of the immune system in order to provide the understanding of the human disease. It's a very important area, and many of you at the institute are interested, particularly in our institute is more on cancer, and cancer immunology is something is very close to his heart. I was just reading in one of those write-up about him. he goes with the passion, and his latest passion is to learn the COVID-19 and the immune response. And particularly, he uses his passion for science to study the autoimmunity. And there are now talks about the autoimmunity and the COVID angle. I'm sure he will probably highlight some of those areas and his recent findings on his laboratories on the work on the COVID. And over to you without wasting we have already lost some of your precious time. Sorry for being a little late, but over to you, Dr. Pillai. We are really happy that we got you. Thank you. Over to you.
Thank you, Soumitra, and thank you all for the kind invitation. I certainly look forward one day to eating shingaras and, kosha mangshu with you sometime. Okay, sure. I'm going to share my screen. Let me just pick that one out. And so 1 second. I think you can see my screen now.
Yes, sir.
Full screen.
Okay, all right. Maybe I have to switch my displays. Yeah, is it okay now?
It's fine.
Yeah. Okay. So I'm going to talk, as Dr. Das mentioned, something about COVID-19, but I'll also talk a little bit about the regulation of lymphocyte development linked to this process. And obviously, I don't have enough time to cover everything in great detail, but hopefully I will get the general message across. Now, in my lab, there's a lot of interesting directions for us. One broad direction is still to look at cell-fate decisions in lymphocytes, and we are interested in how you regulate the demethylation of DNA, particularly when this is happening at a colossal level, as I'll show you in some of these decisions. When you make a big switch from one cell type to the other, how is this regulated? There is a broad interest in the pathogenesis of autoimmune fibrotic diseases and of COVID-19, which is more recent. Basically, because of our ongoing interest in looking at disease, it was an easy switch to study COVID just because there was so much of it around over here. And another thing that we do, which is in keeping with maybe your broader interest in genetics, is that we do have a large cohort associated with our group of single gene defects in humans, which we use to try to understand basic human immunology as well. So many of our studies in the mouse, but a large number of our studies are also done in humans. Okay? And many people in the lab who are involved in this work, I won't be able to describe all of their work today, but essentially some of the studies I will show you today are from Naoki Kaneko and many other people working with them, including Corey Perugino, Jocelyn Farmer, and Vinay Mahajan. And I'll highlight some of Vinay's work as well when we get to it. Okay, and so a lot of postdocs and graduate students who are involved in these studies. I also owe a debt to, to our patients. So we've gone through a great time of tragedy in the world, and it hasn't ended. And, as you know, we are hoping that through science that we will actually have a better day tomorrow, which we are all looking forward to. But I do want to acknowledge that, there are a huge number of people who have passed away from this disease, and we owe them a debt. It's an intellectual debt as well, because we are learning from them and we are learning from people who have the disease today. So I'm going to, have a little tribute, and I call it "The Center Cannot Hold." Immune cells, they tell their stories through signaling cascades. Their tales are never one-sided. A fine balance pervades. What goes up, it has been said, must someday come down. Moderation is the key. There's no need to go to town. Our species, it was naked, faced a foe it had never seen. No antibodies in any humans then to the COVID spike protein. An immune system that is balanced, that generally is the norm. But sometimes immunity goes berserk and the virus starts a storm. Remember William Butler Yeats, but things don't always fall apart. It's when that fine balance is lost that signals go off the chart. Uncontrolled gene expression or a failure of inhibition, and the immune system loses its exquisite precision. That balance, it is crucial. Homeostasis, if you please. The loss of immune precision is the basis of disease. Yeats said, things fall apart. The center cannot hold. Unregulated COVID immunity has sadly culled the old. So most of you are aware of the SARS-CoV-2 virus. I won't belabor this. You're aware of its RNA genome. You're aware that you have binding to ACE2 on the host cell. So you have this trimeric spike protein, and these domains, the RBD domains, can be in the up or the down conformation. And it's when they're in the up conformation that they can bind to ACE2. And you have these conformational changes that occur with different variants at different rates. And this is some work from my colleague Jeremy Luban showing you this. And you also know that once the virus binds to the host cell and binds to ACE2, that there is a protease, TMPRSS2, which will cleave the spike protein. And then you have the fusion peptide exposed, which is basically there's like a spring, which is going to allow the fusion of this enveloped virus, of the membrane of the enveloped virus with the host cell, and you're going to have the release of the RNA genome into the cell. Okay? This is all stuff that I'm sure you're all fully aware of. And you perhaps also know that without an immune response, you actually won't have disease. So, I mean, while we need immunity to protect us, it is also immunity that actually causes us to get severe disease. So think about a healthy lung, you have alveoli, the alveoli have nice air spaces in them. This is what it would look like if you looked at a lung and you looked at a cross section. But when you look at a COVID lung, of course, you have this terrible, inflamed state. The alveoli are filled with fluid. You can imagine why someone would be struggling to breathe with severe COVID. But without the inflammation, there would be no disease. Now, just as I'm not sure how much immunology everybody is aware of, but just we divide immunology in the immune system into innate immunity, which is immediate, looks at generic shapes, and adaptive immunity, which is slower and involves lymphocytes. So innate immunity, immediate responses because you're going to see something that is common to many pathogens. Now, we divide innate immunity into two broad categories again. We have inflammation, and you all understand what inflammation is. I just showed you an image of inflammation. But the second part of innate immunity, which is like a separate function, is the antiviral state. So in SARS-CoV-2, what you want is to have the antiviral state. You don't want to have severe inflammation. The inflammation is, very useful maybe against fungi or bacteria, not that important against viruses. It contributes a little bit to clearance, but not as much. And in this case, we're going to see in the disease, inflammation is unfortunately a problem. Okay, again, if you're thinking of the antiviral state and immunity against viruses, innate immunity, the antiviral state is mainly dependent on type 1 interferons being produced. And type 1 interferons can be produced by infected cells, and they can also be produced in very large amounts by plasmacytoid dendritic cells. And some other innate cells like NK cells can also help with the killing of infected cells. So in innate immunity, we have both PDCs, plasmacytoid dendritic cells, NK cells, and a lot of type 1 interferon playing a role. Adaptive immunity, as you know, is either you make antibodies, and the antibodies protect you from infection, or once you're infected, we rely largely on CD8 cells, cytotoxic CD8 cells, to kill infected cells and to dispose them. And that's how we generally clear viral infections. Now, one of the issues, of course, with having, and since you're interested in cancer immunology, at least some of you are, so cytotoxic CD8 cells had along the way, I just want you to have this thought in your mind, they have the ability to clear off a viral infection incredibly rapidly. If you assume you have a hepatitis infection and you have an intense CD8 T cell response, you would clear the virus, but you would also maybe lose your liver. So you might actually have organ loss. So somewhere along the way, the immune system has evolved the ability to kill and then have exhaustion, where the cytotoxic T cells, after killing to some extent, get activated in a form that allows them to no longer kill, though they can recognize. And this becomes an issue. So exhaustion evolved so that we preserve our organs with severe infections, intracellular infections. But exhaustion is an issue in cancer, as you all know. But this is going to be an issue to consider when we think about COVID-19 as well. So just to again remind you, there's many sensors that are used in innate immunity. We have toll-like receptors, which are endosomal on the cell surface. We have receptors that see nucleic acids in the cytosol. So for RNA, we will think of RIG-I and MDA5 as the key ones. And we also have DNA sensors in the cytosol. Okay? So SARS-CoV-2, of its genome, a lot of its genome is directed towards preventing the induction of type 1 interferons in infected cells. And the virus is designed to evade innate immunity as it applies to the type 1 interferon system and the antiviral state. And that is one of the issues we face. I'm just giving you a couple of examples from the literature. So as you probably know, coronaviruses can use some of their genes, including NSP3, to cause the formation of these budded vesicles that come off the ER. And the virus replicates within these vesicles. The virus can make pores in the vesicular membrane so that its RNA can also come out. A very interesting structural pores that have been described. The other thing the virus can do is it wants to evade these sensors, RIG-I and MDA5. And you would think a viral RNA should have a 5' triphosphate. It has no cap. It should activate these sensors. So RIG-I needs to see the 5' triphosphate. MDA5 is blocked by one of the aspects of the cap structure, which is the 2' O-methyl ribose, which is on the first base, just after the 7-methyl guanosine. So basically, we have these sensors looking for viral RNAs, but what SARS-CoV-2 can do is it has a couple of its genes. Well, actually, one of them is putting the methyl group on the guanosine over here. Another is putting the methyl group on the ribose. So it does all the things that it needs to make its own RNAs look like our messenger RNAs so that we don't respond to it and, the virus evades us. So this is one of the issues to keep in mind. Now, what happens in mild COVID? For many people used to get mild COVID, is less common now. Even young people are getting more severe disease these days. But we assume then there's some data for it to say there's a small viral inoculum. The burden of the virus is relatively low. The antiviral state is still favored over inflammation. So if you imagine you infected this cell in an epithelium, this cell, of course, will not turn on type 1 interferons. But what will happen is that plasmacytoid dendritic cells will intake the virus and produce type 1 interferons, and this will protect neighboring cells from infection. And so you can imagine that in a very mild disease, due to a number of circumstances, including the initial inoculum size and the absence of pre-existing conditions and so on, you do get type 1 interferon production from PDCs and protection of cells, and therefore you clear the virus. Okay. Now, in severe COVID, the situation is a little different. You have inflammation that is out of control. So this is tremendous inflammation, and the antiviral state is now largely evaded. So not just in the cell that's infected, but in a more broad sense. Okay. So this is just from some of our data to show you that, how do we measure severe inflammation? So one of the measures here is we're looking for citrullinated histone H3. And this is a measure of netosis, because when PAD4 is turned on, that's when you get netosis of neutrophils. And you have heavy netosis of neutrophils, even late in disease. And these are in patients who didn't make it. You see the severe increase in netosis. And when we look at ASC1 specs, this is when the inflammasome forms. So the inflammasome is like this little machine that forms and helps to process IL-1, so pro-IL-1 into IL-1, activating caspase-1. So that measure of this ASC spec formation in macrophages also, you see it heavily increased in severe disease. and it increases late in the disease. So there's some interesting aspects of why this is happening late, and we can get to it later if you have questions. But I mean, this is just a measure. There's a lot of inflammation in severe COVID, and we know that. Now, why do some people get severe disease? And we don't fully understand this. Age is a factor. Obesity is a factor. Male gender is a factor. It's not that you don't get it if you're female. It doesn't, in general, obesity does lead to more severe disease. I would argue that we still don't fully understand it. What we do understand, I'll mention briefly. Yeah. So one of the interesting aspects that came out from Jean-Laurent Casanova's lab is a couple of papers showing that people with severe COVID and not people with mild COVID actually carry variants or mutations in the type 1 interferon pathway. And so he showed that, if you add this up, about 4 to 5% of people with severe COVID, but not with mild COVID, have the mutations in the type 1 interferon pathway. What was a larger number, and this may be even larger than what was first published, was that about 10% of people, and this tends to be in older men, 10% of severe COVID patients have autoantibodies, so they have broken tolerance, and they tend to break tolerance against type 1 interferons more in men, more late in life. And they have these autoantibodies to type 1 interferons. And this, in his original paper, it's about 10% of people with severe COVID. So now you can imagine if you have all these, first of all, you can't make type 1 interferon properly even in a PDC. Secondly, you have autoantibodies against type 1 interferons, then you're going to stymie this type 1 interferon antiviral state response, and that's going to make the disease worse. So this is something that we sort of understand at least. Another thing that we discovered, so a long time ago, when I say long time, it's a few years ago, we'd shown that PDCs actually come from lymphoid-like progenitors. And there's others who have shown the same thing, including Florent Guinaud's lab, that PDCs come from these lymphoid-like progenitors. And what we find in severe COVID is that most lymphoid-like cells, including many innate lymphoid cells, some selected T cells, some B cells, NK cells, or they don't expand, they decline. If anything, they decline, during disease. And we showed this in some of our work last year, that we find that if you looked at the transitional B cells, the earliest B cells that come out of the bone marrow, in severe COVID, these cells are not there compared to people without disease. So we're looking at convalescence where they're getting back their transitional cells, whereas in severe COVID, this is gone, just to point this out. So we are losing the cells that are going to help us, both in terms of some of the adaptive responses, at least transiently, and we're also losing the cells that can help us create the antiviral state in severe disease. So just to give you a sort of broad overview before I come to some of our data, is that in COVID, the way you can think about severe disease is that there's increased inflammation, there's a loss of balance. There's increased inflammation, there's less of the antiviral state. Myeloid cells are increased, but lymphoid and lymphocyte-like cells are decreased. Pro-inflammatory cytokines go up. This is IL-6, TNF, and some chemokines as well, but type 1 interferons are reduced. We have increase in complement activation, some spontaneous complement activation, a lot of netosis, coagulation, all of this being driven in severe COVID, and you're losing tissue integrity, organ function, and so on. And in some ways, we can try to explain this is how you get severe disease. Now, there are other questions, One is, how do variants develop? The number of variants, so the world is now, in the throes of the Delta variant, which, did develop in India, almost certainly developed in India. The Lambda variant is from South America is also coming up, but Delta seems to have much higher transmissibility, as does the Alpha variant. So some variants are much more transmissible. How does this happen? Why do we develop variants? What is the mechanism here? I mean, especially when you think about the fact that variants are developing not necessarily in immunodeficient people. So how do they develop? So one of the things to consider is coronaviruses have excellent proofreading. So SARS-CoV-2 actually is doesn't have that high a rate of spontaneous mutation. It's not like, a retrovirus, for instance. This mutation rate is relatively low because proofreading is good. However, the virus is going to compromise host adaptive immunity. More importantly, what I'm going to try to show you is that the host ability to evolve, so the immune system responds, but it does not evolve the way it should optimally, and we will discuss that. This is defective. The virus makes it defective, and it's not the only virus that does that. Severe viral infections seem to make this process defective, probably for an evolutionary reason, which is to our advantage. But as a result, if we can't evolve, then it makes it easier for the virus to go through antigenic evolution. So, I mean, a simple study that illustrates this in a way is that there was a study done in the UK on a patient who had, had COVID but was immunodeficient. He had some variants develop, but the variants gave no advantage as far as we know at that point. This patient then received a plasma transfusion from, convalescent plasma. So he got antibodies given to him. The antibodies are not fantastic when they come from patients. And then he started to evolve variants which went through antigenic evolution. So suboptimal immune pressure is what is seemingly driving the evolution of variants. And the suboptimal immune pressure in most people, we believe, is happening because of this effect on the immune system that's preventing our own evolution of the T-dependent B-cell response. And that's what I'll get to. Okay. So just to recap, we know that antibodies can coat a virus and prevent entry. So this is neutralization. And there is no question that in SARS-CoV-2 infection, in severe infection, for instance, you do get neutralizing antibodies. But the neutralizing antibodies fail to show a property which is affinity maturation. They do not evolve. Okay, I'll discuss that in a minute. And this is just an image of what would happen if the antibody coated the virus. Now, a key process that happens during the development of an immune response is called the germinal center reaction. And in the germinal center reaction, this is a Darwinian process. And this is the secret to our surviving, as vertebrates against all these viruses for, millennia. One of the secrets. The one key event here is that we are going to go through genetic diversification. So basically, an activated B cell, which is helped by a T cell, is going to start to mutate. It's going to divide at an incredible rate, cell division occurring every 2 to 4 hours. And as it divides, the hypervariable regions of the antibody genes, both heavy and light chain genes, are going to accumulate mutations. So you can imagine some of these cells are going to get a lot of DNA damage and they're going to die. But as these cells accumulate mutations, some of the mutations are going to improve the affinity of the antibody. So it's basically the antibody genes are being mutated. The affinity of the antibody for the original antigen, some of them are going to be incredibly better and some are going to be worse. As they go through genetic diversification, the next step of evolution is natural selection. So each of these cells in what's called the dark zone is sent to the light zone. In the light zone, in the germinal center, it is tested. So there's this cell called a T follicular helper cell that actually sets up the germinal center. And the T follicular helper cell is going to give help only to the B cells that can see the original antigen much better than before. So if your KD originally was 10 to the minus 6, and let's say it went to 10 to the minus 9, then you may have an advantage. The cells in the neighboring cells, the neighboring B cells, which come out of the dark zone and don't have a high affinity receptor, are not selected. So there is a very interesting and well-studied process now. Not all the details of which are known, and many people are interested in this, where these cells will go back and forth repeatedly. Only if they're selected will be sent back to the dark zone to mutate again, and then this goes through many cycles. And then in some early on, quite early, after this very limited mutation, some cells will become memory B cells. Okay, but later on, after many rounds of mutation, the cells will then be immortalized as long-lived plasma cells. These will migrate to the bone marrow, and they'll make antibody for life. And what I'm going to show you is that in COVID-19, in severe disease, and mild disease, you don't get this at all. In severe disease, you don't have germinal centers. You lose germinal centers. You can't make this process of evolution. And you will recover this in some percentage of people many, many months later during convalescence, okay, when you have taken away the factors that inhibited the formation of germinal centers. Now, this is some data that we had. So early in the pandemic, so here at the Reagan Institute, of course, we are, there's a lot of interest in viruses. My lab was more interested in autoimmunity, but we realized that we have tools at our disposal to actually study disease rapidly. And so we worked closely with the colleagues in pathology. We realized ethically there's no way we can biopsy any patients. There's no way we can look at their lymph nodes. You cannot study germinal centers in the blood. You have to look in the lymph nodes. We didn't even think about that. We have said, no, it's too risky to even think about it. But we did know that we would have patients who would unfortunately pass away. And we planned and worked with our colleagues in pathology, particularly Bobby Patera, who I teach with for many years. So we worked out that some of my colleagues interested in doing the single-cell RNA-seq, our lab, and a few other labs, we got together and we had this rapid autopsy protocol so that soon after a person died, they were consented, the family was consented, and an autopsy was done quite rapidly, and we were able to look at what's happening in the disease. Now, as I said, we owe a huge debt to all these patients because essentially we are learning from them. So what I'm trying to show you here is that if you this is somebody who died in his 70s, had a rapid autopsy, but did not have COVID. And you see beautiful germinal centers. We're looking at the transcription factor BCL6 in gold over here. So you see the nice formation of germinal centers, and you see this is what you would expect to see. So the normal thoracic lymph node is always exposed to microbes that are coming into the respiratory tract. This is normal, and there are lots of germinal centers. In severe SARS-CoV-2 infection, we don't see germinal centers. Okay, then when we start to look at this in more detail, we're looking at BCL6 positive cells. We're looking at AID positive cells. AID is the gene that drives the mutational process for somatic hypermutation. AID is active. There's AID in disease. Early and late in SARS-CoV-2 infection, we can see it. But BCL6 positive B cells, we're looking at B cells now, are incredibly uncommon. And there's no well-formed germinal center. This is very late when we think the germinal centers are trying to come back, but again, I'll show you the quantitation in a minute. And this is the control. Okay, this is just some quantitation. We're able to actually count cells and quantitate them in tissues. So if you just looked at total B cells, there's a decrease in total B cells in COVID. There's a decrease of total T cells. This is all in lymph nodes. When we looked at BCL6 positive B cells, these are germinal center B cells, and looking at absolute numbers, the absolute numbers have declined in diseases. COVID, these are our controls. When we look at the proportion, now taking into account that there's some lymphopenia, even when you look at the proportion, the BCL6 positive B cells are markedly decreased in severe COVID. And when you look at AID, of course, because of lymphopenia, it looks like there are less AID positive B cells, but in actual fact, if you look at proportions, it's not down dramatically. So T cells are activating B cells, but the B cells are not able to form germinal centers. Okay, so this is a big picture view I'm trying to summarize based on multiple lines of evidence. One of the reasons why these cells might have been gone is that the T follicular helper cells, the cells that should drive the formation of these germinal centers, may be reduced. And BCL6 is, again, it's a transcription factor in germinal center B cells. It's also the transcription factor. It's actually a repressor, but it's the transcription factor in T cells, CD4 T cells, that are going to drive the formation of germinal centers. And this is BCL6 in the nucleus that we're looking at, and we see a marked reduction in COVID of these BCL6 positive B cells. And you can only look for them in the lymph nodes. Okay, so again, we're using different markers. ICOS is one marker, BCL6 is a marker for T follicular helper cells. And you can see that in early and late disease, these cells are reduced. We can see in this case, again, we're looking at the absolute numbers, they're reduced. So using both markers. And when we looked in the spleens of patients as well, there was a reduction as well. Okay. Now, what about antigen-specific B cells? So working closely with some of our colleagues who are using, different versions, they're making RBD with different colors attached to them. So we have 2 flaws attached to RBD. So basically looking for cells, B cells, which are specific for RBD, and we're looking to see that both the flaws light up. So this is, more likely to be a true antigen binding B cell. And then we look at these B cells, and then we, using 25 color flow, we go and ask what populations are they in. I'm not going to belabor you with all these populations because this is a highly nuanced understanding of B cell biology, which I don't want to get into with you today, but just take it for me that activated B cells are all now SARS-CoV-2 specific. Remember, we're looking only at RBD. We looked at spike after that, and there's almost all these B cells that are emerging are actually SARS-CoV-2 specific, and they're in all different categories of cells. Okay, so there's a reduction in the B cells, but we have antigen-specific B cells, including B cells that we consider to be pathogenic, B cells that are linked to disease, B cells that may be activating T cells that can cause damage to our tissues. And that's one of our interests in what we're looking at. Okay. So why are we losing these germinal centers? And again, I can discuss mechanism with you because we are very interested in this. But there is some evidence from mouse studies in severe infection models in the mouse that TNF, a lot of TNF made in the lymph node, TNF alpha, can actually inhibit the formation of germinal centers. And if you actually use TNF blockade, you can restore germinal centers. And we find in patients with severe disease, they have a lot of TNF being made in their lymph nodes. Okay, so there's a lot of TNF. There's some other changes that are happening. So essentially, the milieu of the lymph node is being altered by a severe inflammatory disease. And as a result, we are getting inhibition of the formation of germinal centers because T follicular helper cells are failing to develop. So sort of a big picture view that I can give you is if you normally had a standard infection, I'm using the term loosely, without the issues that we see in COVID, you would have the generation of different T cell subsets. You would activate CD4 T cells, which would be helped a bit by B cells. And these cells will then differentiate into what we call true TFH cells or germinal center TFH cells, which express BCL6. And this will set up the germinal center response. And this will allow the evolution of the antibody response so that we can go from a KD of something like 10 to the minus 6 and end up with a KD of something like 10 to the minus 13. And without germinal centers, we would not make protective antibodies against measles or smallpox or any disease you care to think of. Fortunately for us, this process will be preserved in vaccination, at least the vaccinations we've studied. So this process is okay in vaccination. This process is going to be defective in COVID-19. So in severe COVID-19, there's some increase in Th1 cells, and I'll tell you another cell type that's increased a lot, but there's a lot of TNF being produced. There is a block in the differentiation of T follicular helper cells, and as a result, we do not form germinal centers. We have a lot of these weird B cells, we call them double negative B cells, which we link to disease, which are formed in lymph nodes, which are going to go into the lungs, and they're going to cause some pathology. Okay. So what are the consequences of defective affinity maturation? So affinity maturation is the process by which you go through Darwinian selection and you mature the affinity of an antibody response with time. And so what happens if you don't have a good affinity measurement? So one of my colleagues, Jonathan Abraham, his lab has shown that you lose neutralization breadth. So if you have COVID, you will make antibodies. They will be able to neutralize, but they will not evolve and they will not have breadth. So they will not see, variants or changes that happen in the antigen of the virus. So there's a limitation. And the other thing is they are not evolving and they're not putting enough pressure on the virus. So you can imagine how the virus can now evolve since you're not actually at your best. What about viral persistence? The virus actually, we know persists for many months after you're infected. I mean, there's some clearance from the lungs. It reduces in the lungs, over weeks. But we can find, people are finding virus in the gut, persistence of virus for some time. Why is this? Is there a defective cellular immunity? So we asked this question. And because we were able to look at lungs and lymph nodes and the blood all together, we start to ask this question a little bit. So one of the things we found is that CD8 positive T cells do not expand in COVID lymph nodes. It's kind of shocking to us that, in people who have no infection, the controls, you have actually as many or more CD8 T cells as people with a florid viral infection in the lungs. Okay, so there's no expansion of CD8 T cells seen in the lungs or in the lymph nodes, and I may or may not be showing you that data as well. What we do see is that these cells are somewhat dysfunctional. I'm showing you some measures of this. First of all, there's no increase in granzyme B positive CD8 cells. So the cells that can actually kill are going to have turned on perforin, granzyme B, granzyme A, and so on. And there's no increase. It's like somebody who had no infection. Okay, there's nothing really happening. But what we do see is a lot of the CD8 cells express PD-1. This does not prove they are exhausted, but it's definitely a marker associated with exhaustion. And there's enough evidence to say that this is likely the case, that CD8 T cells in severe COVID are now dysfunctional. They don't get activated, they don't express granzyme B, and most of them have this marker of seen often in exhausted CD8 cells. Okay, so you can imagine, again, we've evolved this process. We had this severe viral infection. We started to kill the virally infected cells. Then we said, no, this is too dangerous. And we are going to go to the process where the CD8 cell is now going to go into the exhausted state. Okay. Although there are relatively few granzyme B positive CD8 T cells in the lung, they are actually a pretty good number, matched in number by CD4 T cells that express granzyme B. Okay, these are cytotoxic CD4 T cells. Now, again, our immunology textbooks, including the one I'm part of, we don't mention these cells much, but many of us study them now because we think they're very important. But we can see now that in this disease, you're going to get an increase in CD4 cells. So this is these are CD4 cells in black. They're going to increase in severe disease. And this compared to non-COVID patients who have very few, in COVID, you're going to have many more CD8 and CD4 cells, which are granzyme B positive. So there's an increase in granzyme B positive CD4 T cells with this marker called SLAMF7 in infection. Okay. So now this is just to quantitate that some more. We are looking at SLAMF7 as a marker of the cytotoxic CD4 cells. And you can see there's an increase in COVID, especially in late COVID, of the cytotoxic CD4 cells. Here is another way of looking at it with percentages. Again, you're seeing an increase in cytotoxic CD4 cells. So these are CD4 cells with SLAMF7. They also have granzyme B, granzyme A, and so on. Okay. So again, the way, just a teleological way of thinking, you should hold my feet to the fire on mechanism here, which we're interested in. The teleological way of thinking is what's happening here is the CD4 T cell that is evolving into a completely different cell type. Instead of becoming a T follicle helper cell, it's becoming a T cell that can kill, but it can only kill cells which have express HLA class 2 or MHC class 2, and that will be cells in which there's inflammation around it where you induce MHC class 2. Okay. So one of the possible scenarios we see is that normally with infection or vaccinations, you would see the induction of TFH cells, and then you see nice German centers forming. But in severe COVID infection, what we're seeing is there's a block in TFH development, and there's what we think is an immune deviation event where these cells are being induced otherwise to actually differentiate, perhaps in a TNF-dependent manner, into cytotoxic CD4 cells. It's almost as though the immune system says, at this point, you're severely infected, our CD8 cells are going to be quiet now, we need something else to start to clear the virus. It won't be as efficient, but at least it'll do the job. And this is what we see. We see a lot of cytotoxic CD4 cells and a deviation away from TFH cells. Okay. One of the reasons why the CD8 cells might be non-functional is that there's actually a lot of TGF-beta also made in the lymph nodes. The milieu of the lymph node is very TGF-beta high. Again, this is a descriptive phenomenon. We're telling you there's an increase in TGF-beta. Maybe this has got some mechanistic issues to think about. Okay. So just to summarize some points, SARS-CoV-2 has a low mutation rate, but viral variants are a major issue. Antigenic evolution requires adaptive immune pressure, but it's probably facilitated by suboptimal human immunity without affinity maturation, and maybe also by dysfunctional CD8 cells. So you don't clear the virus that fast, it persists, it mutates, and the pressure is not enough to clear it, so it ends up evolving. Unlike measles, for instance, there's viral persistence. Natural immunity sterilizes very slowly. Vaccination allows affinity maturation, that we know. It prevents viral persistence. Even if you got infected after vaccination, this has been studied, actually you clear the virus pretty fast. The load drops rapidly. And, it probably prevents viral evolution. If you don't have virus replicating, you won't get evolution, okay? So again, big picture view, in normal infections, you get affinity maturation, high affinity antibodies, robust CD8 T cell response, clearance of the virus happens early enough, you don't damage your organ, you're fine. In severe SARS-CoV-2 infection, you block TFH development, you deviate your CD4 T cells more towards CD4 CTLs, maybe to compensate for the lack of CD8 T cell expansion. Your antibodies are not affinity matured. They actually lower affinity. And not only that, they don't evolve. You do have increased TGF-beta, you do have increased TNF. There's limited CD8 T cell expansion, and there's cytotoxic CD4 T cells going to the lung. And you can see apoptosis that might be linked to the cytotoxic CD4 cells. Okay. Just in the last 5 minutes or so, I'm going to talk a little bit about how we're looking at some of these cell types. We think CD4 CTLs maybe controls the viral spread. They would target class 2 expressing epithelial and endothelial cells. They seem to come to the fore in the context of inflammation and when there is some CD8 T cell exhaustion. We have shown before they secrete IL-1 beta and TGF-beta. They can probably induce fibrosis. They can exacerbate inflammation in COVID-19. So there are some downsides to having these cells. Maybe a potential therapeutic target in PASC. This is the long COVID syndrome. We don't know. We are looking into that as well to see whether maybe we should be thinking of that. Now, in many other diseases, I'm just touching on this briefly, we have seen that CD4 CTLs are the dominant CD4 T cells in lesions. And so here we're just looking at systemic sclerosis, same thing in IgG4-related disease. CD4 CTLs tend to dominate in the disease tissues. They can, they can cause apoptosis. They can deliver. So in systemic sclerosis, you have endothelial cells being killed, and the CD4 CTLs can even deliver their granzyme A into the cytosol of the target endothelial cell, which is then showing you cleaved caspase 3 and is going through apoptosis. Okay, just to give you some background on this. So we think that CD4 CTLs might be driving different diseases, and they might be problematic in COVID-19 as well. But why you get prolonged damage? Why do you have lung damage continuing for months? It may be linked to this process where we are paying a price. We cleared a pathogen, but we are paying the price of having these activated cells, which might be driving some fibrosis and inflammation. So the broader question, this is what I'm going to end with, is can we use genome-wide approaches and CRISPR to better understand lymphocyte cell fate? I mean, obviously we're interested in how do TFH cells become CD4 CTLs? That's one thing. We're interested in TFH cells in general. We're interested in some B cell populations. Can we actually solve these questions? And in humans, can we use these approaches, then go to the mouse, and then or maybe use CRISPR in an in vitro model? This is the way we generally think about it. So if I just took T follicular helper cells, and if I'm looking at T follicular helper cells, we can divide these. We can start with naive cells, and they can divide them into cells that slowly acquire CXCR5 and then acquire high levels of PD-1. So PD-1 is a marker of T follicular helper cells. Although in CD8 cells, we consider it to be linked to exhaustion, it's really a marker of activation of T cells. So repeated activation of CD8 cells which are exhausted, you see PD-1, but a normal functional TFH cell also expresses PD-1. Okay. So anyway, so you have this differentiation profile, and we're able to take all these different cell types, and then we can use large-scale profiling and say, can we understand the transitions? How do the cells go from one stage to the other? And can we then prove these points maybe by using CRISPR and showing that these are important genes that drive the process? So the kind of approaches we use generally in the lab is, we use cut and run. Nowadays we use cut and tag, use ATAC-seq, use whole genome bisulfite sequencing, RNA-seq, and so on, and single cell RNA-seq, and look at different cell stages, and we try to ask questions. Can we now understand the progression and what are the drivers? Can we solve the question of how differentiation occurs by using these broader tools? Okay. Now, so just to give you an example, so we're looking at different stages from naive, we're going to the BCL6 positive TFH cell. Obviously, there's changes in transcriptome to be expected. The changes in methylation occur early, so whatever changed early happened. After that, there's very little change in methylation. So as you moved to a cell that is about to become a TFH cell, there's already demethylation. So some pioneer transcription factor, helped access certain genes, and then you had demethylation to open up the locus. Maybe that's how you think about it. And when you look at chromatin accessibility, this progressively increases, just as you see it for the transcriptome. Similarly, if I was to look at different, using cut and run in this example for changes in H3K4ME1 or ME3 or H3K27, etc., you can see these changes occur with time. And we can now say, okay, can we, by looking at what changed, which genes got turned on, can we find specific drivers in a stage-specific fashion, and can we test that, and we take this back into the mouse, can we go to CRISPR? Okay. Well, the question related to COVID that we've been asking is, how does a cell that was going to become a TFH cell differentiate into CD4 CTLs? Okay, how does this happen? How will we study this? So we found something quite interesting to think about. So again, I want to remind you that we're thinking of an immune deviation event where cells are changing completely from a CD4 type T cell. It's changing its program to now look like a CD8 cell, a completely different lineage almost, but it's still a CD4 cell. It's still seeing HLA class 2, but it's now going to express granzyme, perforin, RUNX3, that kind of stuff. So it's a change in program. How does that happen? So obviously, with single-cell RNA-seq and repertoire, we can look at categories and we can show, okay, there's a subset of cells which have severe, severe disease, and that's fine. We know about these cells. These are cells we are most interested in looking at, but still, how do they differentiate? How do they form? So I'm going to just leave you with a few thoughts. So we have looked at some major changes that occur during development. So when you go from a pro-B1 cell to a B1 cell is one lineage of B cells. The pro-B1 cell is more like a B2 cell. The B1 cell is a self-renewing cell. It's a cell with stem-like properties. It is very different from a B2 cell. But we know that the switch, all that you need is a new BCR and repeated triggering from self-antigen to drive you into a B1 lineage. What happens? Well, how do you switch from this completely non-self-renewing properties of cells to becoming a cell which is stem-like? Similarly, when you think about CD4 cells, we're looking at a cell that is of one lineage, it's CD4. And then it becomes still a CD4 cell, it's still going to see HLA class 2. It's still the same cell it was originally, but it's now going to change and assume many of the phenotypic characteristics of a CD8 T cell, of an activated CD8 T cell, and become a cytotoxic cell. And so in both these cases, I'm going to tell you that there is a massive demethylation event that seems to be crucial. How is that regulated? How does that happen? Where is the balance between methylation and demethylation that takes you there? These are questions we try to answer in part we've published some of our work on B1 cells, and we have a similar work going on in terms of cytotoxic CD4 cells. So this is from published work of ours from this year from Vinay and colleagues in our lab, showing you that when you go from pro-B1 to B1, there's a huge demethylation event, okay? And this is actually demethylation happening of enhancers. And these enhancers are a little unusual in that they have both H3K4ME1, which you would think of an enhancer mark, but they also have H3K4ME3, the ones that are demethylated. Okay, so these are very unusual enhancers, but they are going to be demethylated, and that's part of the cell-fate decision. And we've made some arguments in this paper, if you look at it, about this balance between TET2 and DNMT3A. DNMT3A is playing a role here, not as purely de novo, but maintaining the methyl state. And that balance between DNMT3 and TET2 is causing this change. But still, that's part of what we look at. CD4-CTLs do something very similar. I'm not going to walk through all the data in the interest of time, but there's a huge demethylation. Nothing like what we see for any other lineage. It's much more like the B1 cells. Okay, so normally, if you demethylate, and if I went from naive to, let's say, TFH cell, I showed you some of that data, you would go from 80% methylated to maybe 78% methylated. I mean, there's a small change. But when you look at CD4-CTLs, you're looking here at the degree of methylation of naive cells, the demethylation. So you're looking at hypomethylated regions, and the degree of demethylation of CD4-CTLs, okay, from two different people. So a huge demethylation event that occurs. And just to give you a flavor for what's going on, we don't know all the answers yet. Obviously, this is of great interest to us. If you looked at RUNX3, for instance, okay, let's look at this is a naive CD4 cell, okay, and this is the RUNX3 locus, and you can see the degree of demethylation that is occurring in cytotoxic CD40 cells. Okay, that this is one of the driving transcription factors. I could show you 100 genes, but I'm going to show you a couple just for the examples. This is perforin. This is functionally very important for the function of a cytotoxic T cell. So the CD4-CTL, if you looked at the perforin locus, here in blue is the naive cell, and this is what it looks like. And I can see this huge demethylation that has occurred in the perforin locus. So you can add this up thousands of times, and there are multiple genes that are demethylated. And it seems like, when a cell wants to change its fate dramatically, it has to do something dramatic. And part of it has to start maybe with demethylation, opening up certain completely new sites and maintaining that demethylation state in order to keep the cell fate and the new cell type. Now, obviously, there are many other issues here. If you are, I'm happy to talk to anybody if you want to talk further about it, but I'm not claiming to have answered all the questions, but we are very interested in, and we pursue these questions deeply in the lab. So while we are very descriptive when we study disease, when we're looking at differentiation and mechanism of differentiation, we try to be a little more mechanistic. So to give you the big picture view of what I've told you today, I've talked about the fact that in severe COVID-19, there is actually a defect in affinity maturation. You don't have germinal centers. We don't get higher affinity B cells. We retain low affinity cells. They are neutralizing, but they are lower affinity. They are probably not also going to last that long, maybe a year and a half or so, if I go by the example of what we know from SARS, from the original SARS infection. There is actually stymieing of the CD8 response. There's an increase in CD4-CTLs. And the suboptimal immune pressure that is being generated in this disease for evolutionary good reasons, to preserve the host's life, perhaps. This also, unfortunately, in the case of a virus like SARS-CoV-2, which is so easily transmissible, is contributing to the generation of the antigenic evolution of the virus and the formation of variants, which might ensure that this virus will be with us for some time unless we very effectively vaccinate the whole planet. I have mentioned some of the people in my lab. I just want to mention that we have other collaborators. Naoki is in my lab. He was the key driver of some of this work. Justin, Corey, Katie, Vinay, all are in my lab. Julie and Shashwan are in neighboring labs, but they work closely together with us on this. Bobby Padera has been key in helping us get COVID samples because he's the pathologist at the Brigham who studies this. And we have other collaborators at the Reagan Institute like Shu Yu and Bruce Walker, Daniel Lingwood, Aaron Schmidt, Matthias Lickerfeld, Gaurav, whose work with him I didn't talk about. And this is something that we work collaboratively, which is one of the pleasures of science, working with others. And I think I'm done, and now I'm happy. If you have time for questions, I'll take them. I have to figure out how to quit. And I hope I'm still on. Am I still on?
Yes, we are on time. Thank you so much. We'll take some. Would you like to take some questions?
I'm happy to take questions. I'm available for the next 17 minutes.
Sure. So I think I will just, there is a question from our founder. Today is the Foundation Day, Professor Partha Pratim Majumdar. Partha, can you just open your camera also? So Sheep can see you. Parthoda? His question is whether there are individuals genetically predisposed to small numbers of germinal centers.
So there are individuals, and these are, of course, very rare diseases, in whom there are mutations in genes like AID, there are mutations in CD40 ligand, a mutation in CD40. There's a mutation that this causes a much wider spread disease, is a mutation in IKK gamma, which is, the NEMO, the part of the NF-kappa B complex. So in all of those cases, you don't get T-dependent B cell activation and a proper germinal center response. Those are very rare genetic disorders which cause very severe immunity. In fact, they can be life-threatening as well earlier in life, because without CD40 ligand, you don't even get cellular immunity to function properly. But that exists.
Okay. So any other questions? I think you have to probably post it in the chat box. And Alpana, can you
Yes, sure. So there is a question from Dr. Saroj Mahapatra. He's faculty at NIBMG. He wants to know, is it safe to think that lymphopenia, positive hyperviremia is distinct from the process of neutrophil or NET inducted coagulopathy? That is, these two are separately regulated by distinct set of factors. Or the same pro-inflammatory cytokines such as TNF-alpha controls both processes reciprocally.
Okay, so the lymphopenia may not be linked to TNF-alpha. I mean, lymphopenia, basically the loss of TFH cells might be linked to TNF-alpha, but even that hasn't been proven in humans. It's based on mouse studies. The loss of the lymphopenia in disease is kind of interesting. No one has fully answered that question, but we think. So one of my colleagues has done some interesting studies, Uli 100. So even if you actually cut the spinal cord of a mouse, you get severe lymphopenia being induced. And most of that lymphopenia, we think even in COVID, is actually because cells are actually not coming out of the bone marrow for some time. And this is linked to maybe very high levels of corticosteroids going up in the blood. So it's a multi-step process, but we don't have a complete answer to why there are specific lymphopenia events. We know it for some things. Okay, so we see even the earliest B cells are not coming out of the bone marrow, but we think in part it's a sequestration event where for a period of time these cells are all held in the marrow and not released. Again, not fully understood. So that is different. So coagulation and everything else, that is a little more complex, but I think the coagulation in part is explainable only in part by the activation of complement. So the spike protein can turn on the alternative pathway of complement. And the activation of complement, there's a linkage between complement and coagulation. And then netosis, which is just because you have so much inflammation and activation of neutrophils. Now remember all the PAMPs and DAMPs, as you have cell death, you have DAMPs being released. They are activating inflammation, macrophages, all myeloid cells, neutrophils are being activated. All of that activation of neutrophils also causes, when you have netosis, and if netosis is happening in a small capillary, this can also predispose you with complement activation, which is linked to activate the same proteases can activate the coagulation pathway. So some combination of netosis, activation of complement may lead in some people to a intravascular thrombotic phenomenon, which is causing, coagulation, hypercoagulation.
Devanjan has a question. Alpana, you are going to read it?
Yeah. So his question is, could it be possible that vaccines we received during the middle of the pandemic could give the similar effect of convalescent plasma therapy and new mutant strains generated?
Yeah, so I mean, obviously these are things that some of us are trying to study because we're interested in variant generation, but I actually think it's a little different. I think the combination of prior infection and at least one dose of the vaccine is probably what will give you the best immunity. So I think during infection, although we don't have, affinity maturation from infection, we do have the generation of a lot of memory B cells, which may not last forever, but at least they're there and they're quite broad. And so I think now if you come with just spike and you activate those cells, you will, after infection, after some time, so you have to wait for some time after infection. So the milieu that was inhibitory for the generation of, blockade of germinal centers is gone. So you come back to an almost normal milieu, your infection is, over maybe for many, many weeks. At that point, if you get vaccinated, I think you will probably get the best immune response. There is some data already saying that the best levels of antibodies that we are seeing, and we don't know how long they've lasted because it's all in the last few months, is in people who were previously infected and then got vaccinated. If you just got previously infected, it's not great. But if you're vaccinated afterwards, then it does help. So I actually feel that probably you won't get more variant generation, that you will actually have good immunity coming up, and that will actually, next time you get infected, you'll clear the virus pretty fast.
So any other questions?
There's one more in our chat box. So this is from Tamil Sarkar. He's our IPHD student, integrated PhD student. So his question is, 10 out of 29 genes were associated with immune response inhibition. Is this kind of behavior common for viruses, or is it a unique feature for SARS-CoV-2?
So by definition, if you take any pathogen, what makes it a pathogen is some intrinsic ability to evade innate immunity. Without that, it would be a commensal, right? So something becomes a pathogen typically. I'm sort of broadly generalizing here, but by the fact that it has the ability to evade innate immunity. Whether you're thinking of bacteria or virus, that is part of the process. In the case, so SARS-CoV-2 is particularly good at, and as are some other coronaviruses, at evading innate immunity, but it's not unique, right? I mean, Ebola evades innate immunity, type 1 interferon system, so many viruses do that. So I would say, yes, any severe pathogen has within its arsenal a bunch of genes that are, I mean, always a bunch of the genes, whether you look at a tumor or you look at a pathogen, a bunch of the genes expressed in either is going to evade immunity. I mean, disease is linked to some failure of immunity always. So in this case, the virus is surviving because, it wants to survive. All it wants to do is to replicate in us. And to replicate in us, it has to prevent its source of, division, the cell it infects, from killing it. And that's all it's trying to do. But it is correct. Anything that's highly pathogenic, more likely to have a lot of genomic ability to evade innate immunity.
Okay. There's another question. The question is basically, are we putting all our eggs in one basket? Most frontline COVID-19 vaccines target the SARS-CoV-2 spike protein. This selective immune pressure against the spike protein is expected to generate immune escape SARS-CoV-2 mutants, which will spread globally as vaccinated people will be susceptible. What is your take on this?
Yeah, so I actually think that, I'm not disputing that possibility, but I just think that the vaccines actually do not give you low-level immunity. So I actually don't know enough about the responses to adenoviral vaccines. The little bit we have looked at is that they're not that great. Okay, and I'm unfortunately I have to say that because from what we have seen, they're okay. There's responses, but they're not fantastic. The mRNA vaccines, immunity is very strong. So I actually don't think you'll get enough viral replication to have the generation of variants if you have a strong immunity. Because if you don't establish an infection, you have to divide multiple times in order to make variants. And so the immune pressure is actually good. That's not a problem. Part of that immune pressure is coming from T cells. So now you could say, oh, yeah, but spike alone is not going to be great. We should add to it. And indeed, one of my colleagues, so that's Gaurav Gaiha, who is actually one of my former students, his lab has been creating, and you might see his work, it's a science paper. There's a Cell paper now out on his work with COVID. But basically, they're trying to do very structural design of peptides selected in a way to give you the optimal T cell response. So it is possible. So I think there will be 2 or 3 modalities that will work for us. One, we'll have really good antibodies from vaccines like the mRNA vaccines. 2, there will be vaccines which this may be a combination where while the mRNA vaccines do give you CD8 immunity, so do the adenoviral vaccines. They give you CD8 immunity. It's not that they don't, but there will be vaccines will give you even better T cell immunity to make sure that soon after you get established infection, it gets cleared. So that would help. But the final tool, which is going to come, and it's not going to take that long. I hope in a year we'll have it. When I say that long, I'm talking, in big time differences. I've already seen really good data for protease inhibitors. So the arsenal of drugs that are used both for retroviruses as well as for hepatitis C, for instance, similar drugs are now being designed for the enzymes that are important in SARS-CoV-2. When we have them, if you can now stymie replication with pharmacological means as well, because an oral drug will reach a larger population much faster. I think by reducing the number of people who are having a lot of infection and combining it, you won't eradicate the virus that way, but combining it with vaccination, we will get control one day. And it may never be like smallpox, okay, because this virus is so easily transmitted that it's tough. But I think we'll get control someday. Keep in mind that SARS or Ebola, it's so easy to block transmission because you can isolate people with disease and only they were transmitting the virus. The problem with SARS-CoV-2 is that somebody who's completely asymptomatic can transmit the virus. So we will need to have many tools at our disposal. They will include immunity with high affinity antibodies. They will include CD8 vaccines, so T cell vaccines, and they will probably include small molecules, which are directed towards key enzymes in the virus. And it is this combination that will hopefully bring us back to close to normalcy.
So thank you. If there is no more questions, I would like to show you. Can you see this ship?
Yeah, I can see it.
So it will reach you in next 10 days.
And is the design on it saying NIGMS?
NIBMG, and this is from Vishwa Bangla from Kolkata.
Acha, I'll have to go to a BA Badi to wear this thing.
Yes. Well, next time when you come here, we'll make sure one of the students' marriage you have to attend. that's the easiest thing.
Okay, next time means first time. Yeah, I haven't yet come there, so sometime. Yeah.
Right. Sometime this year only. I think we're planning to have the meeting sometime, so we'll have it. So
probably this year is not the safest thing to consider. I mean, personally, I would tell you that, but yeah.
we'll wait and see how it goes.
I think we'll wait to see how things go. Yeah.
So it's okay. I think over to you, Alpana. I think vote of thanks.
Yes, sir. So thank you, Dr. Pillai, for your wonderful talk and the lovely poetry in the beginning as well. I think in the past few...
So there is a request for him to sing a rap on immune.
You know, if I had time, I will entertain you for an hour, but with different rhymes. But,
so pending.
But I do have a 10 o'clock meeting, so next time.
So Sandeep, over to you.
Yes, Dr. Sandeep, please.
Good evening, everyone. It is my pleasure and an honor to present the vote of thanks on behalf of the organizers of NIBMG Foundation Day today. First and foremost, we are thankful to Professor Shiv Pillai for enlightening us with the research work he has performed in the field of SARS-CoV-2 related immune responses, specifically the role of adaptive immune deficiency in COVID-19 pathogenesis. Thank you very much, Professor Pillai, for taking the time out, especially it is early morning and the busy time for you. Thank you very much, sir. Our deepest gratitude goes to Professor Partha Majumdar who had the vision of establishing an institution of national importance dedicated to the biomedical research using the methods of genomics. His passion and hard work led to the foundation of this research institute. Thanks to the Department of Biotechnology, Government of India, to support the vision of accelerating genomics for health, which is the mantra given by Professor Majumdar to the NIBMG. We are thankful to Professor Sharmila Sen Gupta. Under her leadership, we moved to our present campus from the interim place. We are extremely thankful to Professor Soumitra Das for his able guidance and leadership as the current director of the institute. We are thankful to the speakers of session 1, Dr. Arvind Korwar, Dr. Anup Majumdar, Dr. Devanjan Mukhopadhyay, Mr. Subhashish Ghosh, and Ms. Devashi Tagore. Let's congratulate them for the fantastic work they have accomplished. Foundation Day celebration was made enjoyable and vibrant by the participants of cultural events. We would like to thank the coordinators and all the participants for keeping the event alive and happening. Kudos to all of them, and let's join together our hands to appreciate the efforts. The online events has made it possible to organize our Foundation Day despite the restriction of social distancing. It also has made it accessible across the platforms to whomsoever wanted to view it. All credit goes to our information technology team, thanks to Mr. Bikram Roy and his team members. We also would like to thank all those who are watching us live currently. Organizers are immensely thankful to the support provided by the team members of administration and finance section of NIBMG. Without their presence at the back end, this event would have not been possible. Also, I wish to thank all those who have helped us directly or indirectly to succeed this, to have successful event. But I missed to thank them, mention their names. Thanks once again, all of you, and thank you very much, sir, for joining us. Thank you.
Thank you, Dr. Shiv. You might have moved.
Dr. Alpna, over to you.
So I think we can now. So Alpana, you can close the meeting now.
I think Alpana left it.
I think there might be a failure of Wi-Fi. So I will take the opportunity, sir, to thank everyone once again. Thank you for making this program a success. We were indeed helped by all of you. So now, all those who are in NIBMG campus, we have an announcement for you. Please join us for dinner in the canteen. And all those are on YouTube live and cannot visit us, we owe you a dinner. So thank you very much for participating in our Foundation Day. So with that, I will bring this meeting to a close. Good night. Bye.
Thank you. Bye.
Thank you.
Thank you. Thank you.